seats. There's loads and loads of space because people will be filtering in and out throughout the day. It's an absolute pleasure to have you all here. We've got an incredible lineup of students here and an incredible lineup of speakers here to share a load of wisdom with you throughout the day. Hopefully all of you have got one of these that one of my colleagues has called the Passport of Science for today. So please do bring this with you, fill it up, ask questions, write down notes, do whatever you need to do as we go through the day. As of my last check, we have this streaming at the same time, and we've got members from Brazil, USA, Australia, Canada, India, Pakistan, New Zealand and France all watching at the same time. So it's not just you in here. The YSJ is a properly international venture, so we've got everyone from around the world tuning in. Our chief editor this year is actually in Australia doing her undergraduate degree, but she's asked me to show you this video just to welcome you all to the day.
Hello everyone, my name is Manya and I'm the editor-in-chief of the Young Scientist Journal. I couldn't make it here today because, as you can see, I'm in Sydney, but I thought I'd quickly welcome all of the delegates joining us. On the entire Young Scientist Journal team, I would like to welcome everyone to our annual conference. This conference is more than just a platform to share research. It's an opportunity to exchange ideas and have conversations with your peers. As young scientists, we represent the next generation of innovators and leaders. And so the work we do today has the potential to have a profound impact on the world tomorrow. Thank you so much for joining us again, and we have lots of exciting things in store for you today.
So we've got 5 different guest lectures. And start off with the fantastic Dr. Amy Barker from Oxford Biomedica, I think that's right, coming to speak about gene therapy and her research. So please give a lovely round of applause for Dr. Barker.
I feel like I'm too short to look over this podium. Thank you all for letting me come here and talk today. It's really lovely to see everyone, and I'm really looking forward to the posters later. So if you see me hanging around and looking at your posters, come and have a chat with me. I'm going to talk to you a little bit today about gene therapy, which is what I currently work on. But before I start, I'm going to kind of run through how I got here, what my career's been like so far, because it's been a little bit varied. Then we're going to talk a bit about gene therapy and about CAR-T therapy in particular, a little bit about how drugs are developed, and then we're just going to have a chat about some current issues in medicine, which hopefully will be a bit more interesting. So, who am I? So I live in Oxford, where I work. I'm definitely a cat person. We also have some praying mantises, which are down there at the bottom, and at one point I got to ride a tank, and it was awesome. Which is me personally. In terms of work-wise, I did a BSc in microbiology at the University of Manchester back in the dark ages at this point. I then went on to the University of Oxford, where I did a PhD in tropical parasites. Moved on to spend a couple of years at Novartis working on regenerative medicine, so keeping the cells of the body healthy during chemotherapy. Went to the University of Exeter, to do a postdoctoral position in primary ciliopathies. This is a type of disease that affects people, so it can be anything from lethal before you're born through to just a bit of a loss of sense of smell and obesity. However, one fun thing about it is that one of the symptoms is body axis changes, so you can find that your internal organs are flipped round so that your liver is on the wrong side and everything. I then went to London and did a postdoc in heart disease, and then I ended up somehow back doing the tropical parasites again, and these are African trypanosomiasis. And then after that I came to Oxford Biomedica, decided that what I wanted to do was things that directly led into tested drugs that were going to work on patients, and that's what I do now, so I'm a principal scientist there. What do I currently do? So as a principal scientist at a biotech company, I spend a lot of time, what we actually do is we make medicines for other clients. So people like Novartis, they come to us, they say, we've got this new therapy, we want to use your technology to get it out into patients. So I spend a lot of time talking to them about what is it they're wanting to do, how can we use our technology to help them, setting up experiments to see how their drugs are going to work with our tech. I also manage a couple of junior scientists, so these are people who were sort of fairly early in their careers. I work with various other teams in our company, and we also have sites now in the US and in France to try and figure out how to make everything, all of our processes better, all of our technology work better. And honestly, on a day-to-day basis, what I actually do in the lab is move very small amounts of liquid around. There's a weird echo. This is my team. We are from all over the world. We have everyone from people like me who've done a degree and a PhD to people who came to us from secondary school and are working through an apprenticeship. So the apprenticeship scheme is something that I want to sort of mention today because it's an alternative that's sort of only really started to be brought out in the last few years. And there's a few universities now in the UK that do the apprenticeship scheme for young scientists who maybe don't want to go the traditional university route, and instead you can come in. We've got a young girl in our team who's in her 3rd year now, so she's been working for us, getting paid, and also getting her qualification at the same time. So, my main topic today is gene therapy, and CAR-T in particular, and a lot of newer drugs tend to be biological. And one of the things we've started to do more recently is figure out how we can use the body's own systems to help us treat diseases. Now this is a very, very old video, if it works. So up here you can see the bacteria, the neutrophil, and the neutrophil will chase the bacterium around. This video was made in the 70s on like old school film, and it's still amazing. So the bacteria is trying to escape, that neutrophil is going to chase it down, and eventually it will find it, and it will engulf it. And your body is amazing at getting rid of all these bugs, otherwise we'd be sick all the time. So there it goes, it's actually got it now. And what we want to do is figure out how we can use the body itself to try and cure diseases. And one way of doing that is gene therapy. So in terms of gene therapy, when I talk about gene therapy, I mean altering the DNA in your body in order to make a treatment. And that can be, for example, if you have a genetic disease, we can now actually correct the genes in your cells to put back what you need. So that will be things like haemophilia or sickle cell disease. Or we can do things like adding new functions to cells, which is what we do for CAR-T, where we use the body's own immune system to fight cancer. And there's a couple of different ways, main ways, at the moment for getting these treatments into cells. One of them is to use a viral vector, which is what we do at Oxford Biomedica. And the other one is to use nanoparticles to get proteins and DNA into cells. Now, in terms of gene therapy, it's really very new in terms of our current setting. The very first gene therapy was approved in 2017. So not long ago at all. There was actually one approved in Europe in 2012, but that one got pulled from the market. This is a little bit out of date. This is from December last year. There's been 4 more gene therapies approved this year up to now. So we're at 22 right now, approved gene therapies. 11 of them have been approved in the last couple of years. That number's actually slightly lower now if you take the actual last 2 years. So it's a very, very fast-growing field. We only had 2 approved in 2017, and it's grown a lot since then. This is the list of all the medications, gene therapies currently approved by the FDA. I'm going for the FDA because it's a bit easier. And you can see that there's quite a few at this point for things like cancers, for things like sickle cell disease, for beta thalassemia, so on and so forth. But what you'll notice, potentially, is that quite a few of these are to cancers, and particularly there's quite a lot of CAR-T therapies for B-cell cancers. So about 5 of the list of 20 is B-cell cancers. Now, in terms of CAR-T and what it actually is, in your body, other than your neutrophils and all of those that chase around your bacteria, you have your adaptive immune cells. And the big ones are the B cells. They make antibodies. They're what we target if we're doing vaccines or if we need to make things that will attack extracellular organisms, so things that are hanging around in your body, in your bloodstream, but are not hiding in the cells. And B cells will make the antibodies that attack them. T cells are the ones that attack. So if you've got a virus and it hides in one of your cells, how do you attack that? It's not anywhere that your immune system can see it. So T cells are able to recognize infected cells, and they can go and kill those cells and cure an infection. Now, when I talk about CAR T, what we do is we take the properties of both the B cell and the T cell. So the great thing with antibodies is that they can target almost anything, and they're really specific. So to make a CAR, this is a completely artificial molecule, we take the antibody binding domain that you would normally get produced by a B cell, and we combine it with the receptor that causes the T cell to kill cells. And what we're really doing with this is we're targeting the T cell to kill whatever cell expresses that thing that the antibody recognizes. So we can make these to target almost anything. And in CAR T, we use this to kill cancers. But we don't want to put this, put this gene therapy in every cell of your body, because A, that would be a bit weird, and B, we're still not quite at the point where we can do systemic administration of this. We're still figuring out how to make these things work just in certain parts of your body. So what we do is we take the T cells out, and we make CAR T cells, which are T cells expressing the chimeric antigen receptor, the CAR. Take the patient's blood, basically, get rid of the red cells, keep the white blood cells. We isolate the T cells specifically from that, put in our viral vector or other similar thing to put in our CAR, and then we grow up those T cells, so we've got a whole load of them, and we put them back in the patient, and at that point, they will go out and attack the patient's cancers. In terms of how we get that gene in there, that CAR, what we are using is what's called a lentivector. Now, there are certain viruses, and the classic example is HIV, which are absolutely brilliant at getting into cells, and what they do, which is quite unusual for viruses, is they actually insert their DNA into the host's genome. So they don't just hang around in the cell, they make a copy of their own genome and they put it into your DNA. And that's one of the reasons that HIV has always been so hard to treat, because you've got to not only get any virus that's there, but you've also got to get rid of this DNA, because as long as that DNA is there, that virus can always make more virus. So to make this into a safe viral vector, because we don't want to be giving people with cancer HIV as well, what we do is we take an analysis of the viral genome. We figure out which are the genes in that genome that you really, really need in order to make a virus particle. And the virus particle is really just the empty casing. Everything else, particularly the things that make you sick, we get rid of. They go in the bin. What we know is that it takes 3 of these genes particularly. So one of them is the envelope that goes around the outside of the virus particle. One of them is the Rev, which helps get the DNA from the inside of the nucleus to the outside. And one of them is the Gag-pole that forms all of the structural proteins that make that virus. We can then make versions of these that we can put into mammalian cells. Then we look at our viral genome. I'm just going to go up there. And what we see is that we can isolate the bits that mean that that bit of genome gets packaged into our viral particle. And again, we can get rid of all the stuff that causes disease. So we can make a fake viral genome that the virus will think, oh, this is my genome, I need to put this into my viral particle. But it isn't actually, it's what we want to put in there. We then put all 4 of these things, so our 3 genes that we use to make viruses and our fake viral genome, into mammalian cells, and once they're all in there, they'll all start making proteins and copies of the genome. And what they'll do is they'll assemble that into a viral vector. So this is a standard viral particle with our genome in it, rather than the viral genome. And we can take that viral vector, and we leave all the things that are required to make a virus behind in the cells that we made this vector in. So they're no longer there in that viral vector, which means that when we infect something with that viral vector, it can get into the cell, it can get its DNA into the nucleus and integrate it into the host cell DNA, but it can't make any more virus. All it's going to do is it's going to produce whatever we've put in there, which in this case is our CAR. And that means that if we put this onto T cells, we get CAR T cells. So, as I said, we can make CARs to target almost anything. So why haven't we cured cancer yet? So, there's a few problems here. First of all, if you want to target a cancer, you need something specifically only on that cancer and not anywhere else in your body. And the problem is that most cancer cells come from your body. And there's not very many of them where you have very specific markers that are only on that cancer cell that are not anywhere else in your body. So that is one big problem. We don't want to be killing healthy cells. We certainly don't want to be attacking something that's found almost everywhere. That would be, very bad for you. Cancers are also very hard to attack. So one thing with particularly sort of solid cancers, and one of the reasons why B-cell cancers are so heavy on this list, is that B-cell cancers are leukemias. They're spread out through your bloodstream. The cells are fairly individual. It's very easy to get at. But a solid tumour, something like breast cancer or melanoma, they tend to be very tightly packed. So the problem is that it's very hard to get anything into the middle of the tumour. And that includes even sort of, blood. So you'll often find that the centre of a tumour, a solid tumour, is just dead cells. They can't even get oxygen or nutrients to them. And the last thing is that cancers are really variable. So they mostly arise by mutations. And the thing is that once they start mutating, they're more liable to do more mutation because they're growing so quickly. So they're prone to mutating further, which means that they can then become resistant to whatever we're trying to put into them, which is always a problem. The other drawbacks with CAR-T, the side effects are potentially very limiting. So you imagine we're using the body's own immune response to attack a cancer, but if you've got a cancer, you've got loads and loads and loads and loads of cancer cells in your body. And we put in our CAR-T, and that triggers your immune system, and that can make you really sick, because when you get a virus, like when you get a cold, most of the symptoms you get when you get a cold, they're not from the virus that's infecting you. They're from your body trying to attack that virus and get rid of it. So we can find that you can get really overwhelming immune responses with CAR-T to the point where they can even cause death from the treatment. So that's something that obviously we've found lots of different ways to get around and to limit the responses that we get. A lot of CAR-Ts rely on viruses like the HIV in order to get the gene into the cells, but one problem with HIV is there's no specific part of the genome it will insert the gene into. It will go wherever it feels like. So there are some cases where the anti-cancer treatment, the CAR-T, has caused another cancer. Now, if you're already dying of cancer, this is a very small risk, which is one of the reasons why CAR-Ts are here, and most of them do not do this, but there have been a few cases of early stage clinical trials which have shown this as a side effect. So again, it's something that a lot of companies are working on ways to direct where the lentivector inserts into the genome. And the other big problem, CAR-Ts are very, very expensive. Now the vector itself isn't stupid expensive. It's a biological molecule rather than a chemical, so it's a bit harder to make. It is more expensive than, say, taking an antibiotic. But the main thing is that because we've got to take the immune cells out of the patient, we've got to expand them up, we've got to put them into culture, put the vector into them, test them, expand them, and put them back into the patient. That patient is potentially hospitalized for weeks. This is, a several week-long process to do that involves a lot of skilled labor, a lot of intensive work, and that means that the actual price for a CAR-T treatment can be around £500,000. And that leads us on to the next part of the talk, which is the drug development process. And this will partly lead into why our drugs priced the way they are. So in terms of drug development, you'll usually start off with an initial phase. And that means for example, I want to target this cancer, or I want to target that thing. But just because you know what you want to target doesn't mean you can actually do it. So the first stage is almost always coming up with how can I do what I want to this target. So whether it be I need to create a CAR that will kill this cancer, or whether it is this enzyme goes wrong and I need to find something that'll stick to it to fix it, or whatever it is. And there's a few ways of going through that. You can do library screening. You can buy libraries of thousands and thousands of molecules. You can test them all against your target, see what works. You can use a template, so if there's already something you know of that works, you can take that and use it as a template to create your own drug. You can do structural design, so that's kind of a, okay, we know this thing has a binding pocket at this particular point, so we're going to design something that will go into it. Or you can use computer-aided design, which you'll probably hear a lot more about in the next talk. Once you've got that, you've got to screen it against your actual disease. So you are going to need, whether you like it or not, unfortunately, you are going to need to do animal work. And you're going to need to show that, yes, this works in a Petri dish, but does it work in an animal? And pretty much every drug development agency, every government agency that approves drugs is going to require this. You're also going to need to do some modification. So just because you can affect this target doesn't mean that you can turn this into a pill and someone can take it. You need to know it's going to be safe, it's going to get to where it needs to go in the body, it's not going to degrade 3 seconds after it hits your tongue, things like that. So that can take anywhere from like 2 years to well over 20 years. Once you actually get there, you need to take it into humans. And that, clinical trials, is a whole mess of its own. There are so many stages. You've got to figure out who your patients are, get ethical approvals, register it as a trial. You've got to recruit doctors, because who's going to treat your patients? You've got to recruit hospitals and medical centres, because those patients need somewhere to go. You're going to need to figure out what samples do we need to take. You know, we need to take blood from these patients or tissue samples. How are they going to be stored? Who's going to sort all that out? And the other thing you're going to need to do is prepare some human-grade material, which is a lot fancier than the stuff that we put into animals. So, good manufacturing practice, or GMP, is the main thing we use to define our medicines, and it means that everything has to be very, very, very tested, because we want to make absolutely sure that the thing that goes into a person, first of all, is actually what it says it is, and nobody's picked up the wrong vial at some point in the process, and secondly, is absolutely safe, and we know everything that is in there. And this is regulated by various different bodies. In Europe, we have this thing at the top by the European Medicine Agency. Any manufacturer of medicines for use in the EU, no matter where that manufacturer is based, even if they're based in, in Australia, in China, in the US, if they want to sell in Europe, they have to subscribe to GMP manufacture. And GMP itself is a whole thing. So you start off with your small scale. You may be making, this is sort of the scale I work at, little shake flasks that I can see. I like those. You do your initial analysis. You're then going to scale up, probably do a mid-scale, say this is 10 to 50 litres of cell culture, so it's about yay big. You're going to purify your drug. You're going to do a whole bunch of testing to see exactly what's in it. Is there any protein in it that could affect people if we put it into them? Is there any DNA in there that could somehow affect people? You're then going to need to look at your GMP batch. And for GMP, you need to buy very heavily tested materials. You can't just buy a bottle of whatever off the shelf at the science shop, like we have those. And you've also got to check all of your equipment fits the regulatory guidelines. You've got to do a whole bunch of setting up all your testing to make sure that you know exactly what's in everything. And then you finally go and make your big GMP production, which is 200 to 1,000 litres, which is like that. Purify that out, and then do all of your testing. You've got your sterile vialing there to make sure everything's sterile, because we don't want to put any bacteria into it. And eventually you get to batch release. The absolute fastest I've known this happen with a biological is a bit over a year. More often it takes 2 to 3 to get through this process. And then once you've got there, it doesn't stop. You then go into clinical trials, and that's multiple phases. So your first trial is usually a very small trial, maybe as little as 5 patients. If you've got a rare disease, maybe up to about 20. And it's not really designed to see if your drug works yet. What it's designed to do is to see, is it safe? So you spend 1 to 2 years getting these patients in, checking it, making sure nothing bad happens to these patients, this drug is safe. At phase 2, when you go up to maybe a couple of hundred, up to 100 patients maybe, that's when you start to look at what dose do I need to give? Is this actually working? Does this treat my disease? And then phase 3, that's the big one, over 200 patients normally. This is where you look at efficacy, whether it's actually working, and it will usually be tested against either a placebo, so a fake drug, or against other approved treatment. So basically we're looking at, does my drug work? And if it does work, is it better than what's already out there? Because obviously, especially in things like cancer, placebo trials are not a good thing. You know, we actually do want to be treating these people. You then apply for approval. The FDA takes all your paperwork, all your very meticulous records of exactly what is in this drug, exactly what you manufactured, and they decide whether to approve it or not. And even then, it's still not over, because then you're into phase 4, where your drug is out there, doctors are prescribing it, but you're still looking constantly for, are there any side effects coming up? Now that you've got a really big population, you might start to see rarer things you maybe didn't see in trials, and things can still get pulled. So overall, this can be anywhere from about 10 years at the fastest to well over 30. And at every single stage, many, many, many drugs fail. There's just been a very high-profile one that's been pulled at phase 3 for schizophrenia from AbbVie. So for every few thousand medications that get submitted, only one actually makes it through to being an approved product. So making a drug is a very time-consuming and very expensive process. You're talking maybe 30 years of half a business working on something to maybe get approval. And if we want to keep making new drugs, these companies need money. So unfortunately, some of the price of this research has to be costed into the price of the drugs that we sell. Estimated cost of drug development is a very, very controversial topic, and it varies a lot depending on how well known the target disease is, on what kind of medication it is. So biologicals do tend to be more expensive because they're more time consuming and research intensive to make. Anywhere from 1.3 billion for a drug, this is in dollars because everyone works in dollars, through to maybe as little as 113 million, which is still a staggeringly extortionate amount of money to me, all the way up to over 6 billion, depending on whose research you're reading. So that leads us into my first topic on the what are our issues, and the first one is medical budgets are limited. Here's a question. This is actually from one of the apprenticeship schemes, and I really like it, so I use it. You're in charge of the healthcare budget, say, you know, the NHS budget for a city of 3 million people. You've got £1 million and you need to decide how to spend it. What you can do, here's your options. 2 CAR T therapies for babies with lethal cancers, rare cancers. You can do 100 hip replacements for people who are having trouble walking. Or you can do 1 million courses of MMR vaccine. Now I'm going to say right now, there is no correct answer here. Who would vote for the first option, the gene therapies? Just give me a wave. One down there, a few back there, yeah. Who'd go for the hip replacements? One lonely person at the back. Who'd go for the MMR vaccine? Overwhelming number of you. Okay. Anyone want to shout out and just tell me why? Anyone feeling really brave? Yeah, so the number of people that you can potentially save, you know, 1 million courses of MMR, that's a lot more people than 2 babies. This is the trolley problem writ large. Who'd go for hip replacement? So if you're thinking about this, yeah, 2 babies, it's not very many people, but this is rare cancers. We can get more information maybe to help more drug development. And also, they're babies, they've got a long life ahead of them. Hip replacements. These might be people who are out of work. You know, they can't get around. So you're potentially contributing more to getting people back in work, contributing to the economy, contributing to helping other people. MMR vaccine, I mean, vaccines are a massive thing. They've really helped with so many things. And on the other hand, that's 1 million courses, and you've got a city of 3 million. So how many of your people are already vaccinated there? And that might also come into kind of your thoughts on this. The NHS has a division called NICE, which is a wonderful name, National Institute for Health and Care Excellence, who decide what we're going to fund. And when they look at it, they're looking at the cost and the benefit. Is this worth the money? Does it give us enough? Does this cancer treatment lead to people living long enough to make it worth what we're going to pay? And the other thing I want to bring you, now we're talking about drug diseases and also about the MMR vaccine, is leading causes of death. So on the far side from me there, in the green, that's low-income countries. This side, high-income countries like us. Biggest three killers in high-income countries: cardiovascular disease, heart disease, malignant neoplasms, cancer, and neurological conditions, things like Parkinson's, that kind of thing. Over in the low-income countries, that big bar at the bottom, infectious and parasitic diseases. Then you've still got your cardiovascular disease and neonatal conditions, things that kill babies. Those are the big killers in poor countries. This was 2019 from the World Health Organization. I think there is a new one this year. They release it every now and again. This is the list of things, various different infectious diseases. Again, low-income countries and high-income countries, and this column at the end is the difference between the two. And I've coloured it. It's more red depending on where you get more deaths in the low-income countries. And some of them, diarrheal diseases. Diarrhea is killing people. We don't even think about this. You know, you get diarrhea, you spend a couple of days on the loo, you go to the doctor, you take some Imodium, you know. The other one I really want to draw your attention to, whooping cough and measles. These deaths are multiples of thousands. So that's over 1 million deaths every year in low-income countries from measles, which is something, again, we just don't even think of here. And part of that is because they can't access vaccines. Now, I spent a certain amount of my career doing African tropical medicine, parasites, and as part of that I was lucky to go out and do some teaching at the University of Ghana. And it's very interesting talking to the students there, because you talk to students here and you ask them, you know, what do you want to work on? And it's heart disease, it's cancer, it's things like that, because those are what's killing the people we know and love here. There, they want to work on cholera, they want to work on TB, they want to work on malaria, because that's what's killing the people that they love. So why can't they get medicine? It's not just because they're poor and the countries can't afford the medicines. It's because they can't afford doctors or doctors' training. It's because a lot of drugs aren't designed to travel long distances, not refrigerated in tropical countries. It's because a lot of small villages, they may not even have clean water, let alone healthcare. And there's also, a lot of the time, there's no registration of citizens. So one thing we know, particularly with tropical medicine, is that the numbers we see as to how common these diseases are is probably massively underreported, because a lot of these villages, they never, you never know who's born and who dies there, let alone what they die of. And another problem is that there is still a fear of technology. There are people who are still out there, they're scared of taking medicines, they don't trust the doctors. And one other problem was religious teachings as well. So back during the sort of start-up of HIV, and there was very heavy transmission in Africa, but a lot of the people living there, a lot of those countries are very religious, they're Catholic, and the Catholic teachings forbid use of condoms, which is one of the best ways to prevent HIV transmission, so that can also cause problems. But it's not just out there where people are scared of technology. So the Frankenstein effect. So Dr. Frankenstein made this monster. You know, he created a living being, it was amazing, but everyone was scared of it, even him. And if you really want to, this is the National Theatre Live's production of it, which you can sometimes catch online, where you get to see Benedict Cumberbatch crawling around the floor being Frankenstein's monster. It's great. And the most obvious thing we've had with this is the COVID vaccine just a few years back, and the number of stories that went around about how this was Bill Gates injecting microchips into people, or that it was designed to kill certain populations, or things like that. And there's still a lot of people who didn't get vaccinated, and that causes issues. I mean, we are now having outbreaks of measles and chickenpox in California that are killing children. And we can cure this, but people are afraid of the technology. Another issue we have still, and particularly in the rise of social media, is snake oil. And this is kind of an old traditional poster for, someone would come around the village, they'd sell you this amazing tonic that cures absolutely everything. And always it sounds a bit too good to be true, but, it's what people do. Especially the rise of social media and fear of technology has given rise to a lot of this. So a couple of prominent examples here. This lady, she had a whole sort of social media empire built on the fact that she'd apparently cured her cancer with a specific diet and lifestyle. And she made tons and tons of money off people who wanted to believe this because, you know, chemotherapy is scary, but I can eat healthy food and maybe get better. And then she admitted in the end that she'd never had cancer at all. This family, they believed in homeopathic medicine and herbal medicine, and when their son got meningitis, that's what they chose to treat him with, and he died. And they were actually charged with negligence for not providing him the essentials of life. And this is a family who's been suckered in by one of these snake oil schemes, and it's led to the death of their child. So this is still, and a lot of this comes from fear of technology. A lot of this is, you know, you see these wellness influencers and it's like lifestyle, diet, sunshine, exercise, and yeah, those are all really great things, but if you've got an infection, take an antibiotic. And the other thing I like to say to people who say, well, but it's natural, so is arsenic, you find it in apple seeds, I'm not going to use it to, like, treat my diseases. The really complicated bit is that sometimes scientists themselves are the scammers. So you'll see a lot of these snake oil salesmen, you know, oh, well, this guy's a doctor. It says so. He's got this qualification from this place. Now, I'm a biologist. I know absolutely nothing outside of my field, honestly. You asked me to look at, you know, something I've never worked on. You asked me to look at, I mean, I work on CAR-T, and most of the immune system is still a bit of a mystery to me. So if I try to sell you something about the immune system, you definitely shouldn't believe me. Most of the time, when science goes a bit wrong, you get hundreds of papers retracted every year, published papers. Most of the time, it doesn't affect anyone outside of that immediate field, but every now and again, you get a big case that really has knock-on effects, and the biggest one, while I've been alive, has been the MMR problem. And that was a study published back in the 90s, I believe. About 15 people on this, led by Dr. Andrew Wakefield, who linked the MMR vaccine with autism. And at this point, all of the other authors, apart from Wakefield, have retracted this paper. And he's stuck to his guns, and he's been struck off the medical journal because of it. And even though this has now been completely disproven, this is the myth that everyone still remembers. Because everyone remembers the big controversial case. They don't remember all the stuff that came after it. And this is still causing a lot of fear today. So what I would always say, particularly when you can find scams online, think about it critically. First of all, is this person trying to sell you something? Wakefield made a lot of money from selling stuff to scared parents. If this person's trying to sell you a book or a subscription or, you know, a lifestyle, think about it and go, this person has a vested interest. The other thing is how many people, so I mean, it's like climate change denier. There are scientists who deny that climate change is happening, but there's an overwhelming body of scientists in comparison of numbers who are not. So have a think about, you know, if this one person supposedly has found the magic secret, how likely is that? So, I'm going to finish there. I'm going to be around all day. Come have a chat to me. I want to come and look at all your posters and come and talk to you all, see what you've all been up to. You've got my email address up there if you need anything, you know, some advice or anything like that. I don't know if we've got time for any questions. I think I've overrun a bit. Does anyone have any questions?
Any questions?
Echoing silence. Everyone's too afraid of me.
Can we have a lovely round of applause for Dr. Barker then? Yeah. Thank you so much for that. Our second speaker of the day is Dr. Paschoal, who is going to come and speak about his work in biological data science. Both speakers will be out here throughout the day, so if you want to ask questions, please do come and grab them. But without further ado, Dr. Paschoal.
Hello. Okay, good morning, everyone. Well, thank you for coming, and thank you for the opportunity to be here. Oh, So my talk will be more related with data science and how you could join with biology, that I call biology data science, from discovery to function, from data from function discovery. Before to start, I want to, in the name of Ben and all the institution, the King School Canterbury, thank you very much for the invitation to be here and opportunity to talk a little bit about our work. It's always good to show not only for scientists, but different audience to show and say great things that I think we do and you could do together. So basically, I promise you will be less than 40 minutes. The talk is, I'll talk about very fancy artificial intelligence and biology, how this combination is very fancy and perfect for up to date. You can see the Nobel Prize in the last month, how many work between biology and the artificial intelligence, and of course show some example, not theory, but also in practice. So let's start with this idea. So this, I think everybody play hash game. So in this case here, the human are in blue, the computer artificial intelligence are in red. You can see that you have now is turned off red, the computer, and any situation here, if you, any place that the computer put their mark, the circle, that you lose, but then you see the end of the lecture, what's happened. Okay, so keep in mind this example because I'll give you some ideas and insights, then I'll back to talk about that. So just a brief introduction about myself. So I have done my undergraduate course, Mathematics and Informatics in Brazil, but before that and during this time, I stayed 8 years working in a company, in more programming, internet company, web development. Then I live in the industry and go to academia. So I have done my master in computational modeling in Rio de Janeiro, and then University of São Paulo is the best of Latin America. I have done my PhD in bioinformatics, and later on, after I got position as assistant and associate professor, I have done my postdoc 1 year in Germany. So this is more in bioinformatics. This is my background. So just to show a little bit, Brazil is so huge. So my university that I have been the last 14 years, the first state in the south of Brazil, in Paraná state. So most of people know Rio and São Paulo. And you have 13 campuses spread, the UTFPR, the Federal University of Technology of Brazil, Paraná, sorry, there are 13 campuses spread. I'm one of Cornélio Procópio, the north of the Paraná state, just to keep you in mind. It's more a technological university, it's a young university that focuses more, develop more technological aspect. And in my campus, in particular, I'm from the Department of Computer Science. You have more STEM, engineer, mathematics, computer science, mechanical, electrical courses. If you are interested more in particular group, you have a bioinformatics and pattern recognition group. So that is not myself or the other professor. You go to the website, there is English version. You can go see and more or less what kind of research you go there. And also you have the master and PhD in bioinformatics. I have the opportunity or maybe the challenge to get the coordination of a master degree during pandemic time. That's so different. And the last year, I will complete next week 1 year that I moved now to UK and I start a group lead position, the Rosalind Franklin Institute. In the afternoon, Mark Basham, my boss, my director, I hope I have a good presentation. You say to my boss that I have done a good presentation, please, to keep my work. So Mark, you'll talk a little bit, a different perspective, what you do in Franklin. So the Rosalind Franklin Institute is a national institute in the Howard campus. Just to give an idea, it is 4 minutes, half an hour from London and half an hour more or less from the south of Oxford, close to Didcot. It's funded by EPRCC, the Physics Council. And this is our 4 major values. So you have a venture to focus on project that's very risk, breakthrough science, novelty to create a new technology that is engaging for the society, and of course the utility to provide this technology also for the industry, academia, etc. So these are our 4 values. This is our perspective. You do work in technology, develop technology across all these main fields, since the molecular to organisms and so on. And of course, as I said, I will complete now next week 1 year that I start in the artificial intelligence and informatics teams with Mark will talk in the afternoon for you, Laura and Michelle. So we work in different fields. Michelle in bioimaging, Laura in data management, and more I focus more what I call biological data science. So let's start now. So what's exactly is what I call biological data science? So imagine that you have this sequence. For some people from computer science, just a string, computational string. And people from biology could be a nucleotide, so you have ACGT. And why you have this letter? Because it comes from DNA. And why you have this letter in DNA? Because biology nature wants that, and you have this combination, cytosine, guanine, thymine, and adenine. So this combination, C, G, and T, and A. Why? Because, of course, there is a chemical, I'm not from chemistry, biology, so, but there is an explanation about that, because the numbers of the hydrogen bonds is this combination between T and A, that is 2, and G, C and G, that is 3. So I'm from computer science, but I had to learn a lot of biology, molecular biology, genetics, biochemistry to do my work. And that's one point I want to convince you how it's interesting and important to know the other work. I'm not a biologist, but I need to learn to have this interdisciplinary research. Okay, and of course, just to remind you, what's critical, of course, the Franklin was it gives you the evidence to make it possible to discover and define the structure of DNA. And if you go, you have this, from Franklin, have done this X-ray diffraction experiment. And if you go in our building, you can see the same draw in our building. It's very nice. So let's give you one information. I give you that ATG is when you start the gene, and I give you TAA, the end of the gene. Then I ask you, what is the gene? And you could start to looking, probably, most of you start from the beginning of the sequence, start to see what is the ATG. I will help you. We found here. Perfect. Then you found the beginning and of the end of the gene. I put in an algorithm in a computer, and of course, and do large scale. Okay? And then I will ask you if this sequence will translate to a protein, for example. I found the gene, and then I found the protein. Perfect? Yes, but of course, life is not so easy. We have, of course, alternative start, alternative end, and which one is correct? So which one is giving a different protein, or which one are not giving nothing? This something is start to be more challenging. And of course, if you go to bacteria, is one aspect. If you go to eukaryotes, you have intron, exon, and so on. That's why important to know. Although I'm, again, from computer science, I need to know the biology, etc. Otherwise, you I do things that doesn't work. So take the message, knowledge is power. That, for example, Darby said, is not the most strong or intelligent that survive, most responsive to change. So that's the message. And of course, why? Because otherwise you build things that doesn't work. Okay, so that's, I like to say this, most of people computer science, because people computer science want to do very fancy algorithms, they don't care about the domain of the problem. And when you work in the interdisciplinary, you need to understand the domain of the problem. Otherwise you do that. Okay, it's waste of time, money, etc. So what do you want to do? Why I tell you that? What are your goals? So first you want to discover things, as our first speaker said. So you want to know about what you are doing. For example, why you have so diverse species in our planet, what they are doing, what's the difference. So you know that the different cells, for example, eukaryote cells, that you have the membrane in the nucleus, and the bacteria, there is no nucleus, so the DNA is all inside the cell. You have the same gene in different cell doing different product, like say somehow in this way. What happens? So you have a muscle, you have a skin, you have nerve cells, and different products from the same DNA. That's so fancy, that's so interesting, what they are doing. How is this process controlled, and so on. Looking more in perspective of computer science, when you're looking for the data, the big question of my group and our work is, okay, what is the data telling us? And I like to represent data as a bunch of blocks. I think everybody play with blocks, so you have different type, shapes, and size, and this is somehow represented how is the data you normally work. It's not perfect. Let's give you one more specific example about that. Is more, we call exploratory data analysis. Any more, when you have a data, you want to understand the data, you do what you call EDA, that exploratory data analysis. It's more to understand what was the distribution of the data, what you have in hands. This example in biology is about transposal element, so I'll talk about mobile transposal element. By definition, it's a classical definition, mobile elements or transpose elements are elements that could move or copy the genome, are repeat elements that have this property to move and copy. So if I summarize for the computer science vision, it will be a mechanism of copy and paste and cut and paste. Sorry, the biology, just to make more friendly the definition. Why they are important? Because, of course, I need to say about this wonderful woman. So Barbara McClinton was American plant biologist that in 1943, she proved experimentally that this happened, that is this event that could move or copy. And the people said, no, this is not true, it's bullshit. And just in 1983, 40 years later, she got a Nobel Prize. Just to show how you need to be resilient, how you need to believe in what your ideas and so on. So what the motivation to this work? So we need to think always to show the society why I'm doing that. For example, we know we are facing climate change. This impacts our lands and food and crops. So we are facing crops lose, and you have some percent here. That's impact, for example, I come from Brazil, that agriculture is so important in the vision of economic. And of course, you need to create more tolerance varieties for this climate change. And some plants' genome, more than half of the genome is composed by repeat elements like transpose elements. So it makes sense to study of that. Just to give one more example, each of these cases, the corn, the orange, the shape of tomatoes, the type of grapes are direct or indirectly influenced. Some cases of the influence of the variability of transpose elements. So it's more genetic information to show how it's important based on society and science. So the question here is, okay, let's get the public data. I'll get the data from literature for other groups that publish a database, and I want to see how is the data. So I look in just the distribution of the length of this sequence, okay? Just the size of the sequence. In this case, I choose LTR because it's the most abundant transpose element in plants. And you can see here in blue, orange, and green is 3 different groups. One group is, the first one is the previous team leader from EBI, the European Bioinformatics Institute, here in UK. The second one is a group between Colombia and France. And the third one is a group from here, from UK. 3 different groups for the same class, for the same categories of data, for the same plants. Now, in this case, rice, and 3 different shapes. Which one is correct? If I want to create a machine learning tool, which one I choose and why? Just to show you that life is not easy, okay? It's public data in literature. You can get what's published, etc., etc. So take the message, do not believe the data, even for the great institution, because as I said here, you can show you, Prizegiving said I have 3 kids, not the opposite, otherwise you make a mistake. So be careful, even wherever comes the data, do your analysis from the raw data to prove that you do not have any mistake. So the message here, the second message, is not trivial to work with biological data, okay? So let's show some challenge and then one more example how this connection between artificial intelligence and biology. So in my opinion, there is 3 key main challenges. The first one is how to get the data, and you have some example now in the first talk about gene therapy. So, and is wet lab, that's something I don't know, I just know to go to the computer science, but to get the data is so important. The second one, the quality, and so related with the first one, because if I do not work well in the first step, I do not have good data. There is a very fancy professor, is called Andre N.G. from Stanford University, from deep learning, and he has a talk, he gives kind of this message. If you work to improve your model, you could get up to 20% in performance better. But if you have a very nice, good quality of data quality, then you could get up to 80% in performance, just to show how it's important. And of course, after you have good data and you start to do the analysis, what does data tell us? What's the answer? What's behind the scene? So, as I like to show you, consider data as a bunch of blocks, different shapes, sizes, and format. Data is crucial, but itself only is not enough. So, what you try to do in our work, in our lab, is to look very carefully with this data and try to prepare, pre-processing, data visualization, exploratory analysis, and of course, then you could understand the data and decide the distribution, and then build the house, build the machine learning, build the tools, etc., do prediction model, etc. Of course, I'm not a biologist. I do interaction with the biology, life science, etc. So my work is the bridge between the biological problem and the validation. We are discussing in the morning, so I give the best, probably 10 candidates or 2 candidates to go to validation when you have 3 million candidates. That's the idea. And of course, there is some challenge. 3 men here. The first one is interdisciplinary background. As I said, although in computer science, I need to learn more about the other science to understand the domain of the problem. Human resource, we need people, and we need good people. You need you. So what you learn, you just discover how is important what you learn after you start to work and do, if I focus more what that teacher was explaining to me, now I need to study again what they said. And of course, think that you're using different aspects. For example, in my work, I'm using sometimes data integration, I build a model, or just simple statistics analysis and so on. And why you are doing that, for example, why I get the data and apply artificial intelligence or data science? Because at the end you want to guide the decision making. As the example of the previous speaker, you have 3 situations, which one you are focused to do your investment of your money. You have 3 options, but you have money for just one. So how you are going to do vaccine and spread in UK to face against COVID-19. So it's a timing, money, resource, etc. So the idea is that the artificial intelligence will help us to provide more information to guide the decision maker. That's the main focus. Again, and the big question is that, what does the data tell us? In my case, when I said what the data tell us, is to extract insight. With insight, what's behind the scene was not so clear. That's why we apply computer science, because I don't like the idea of artificial intelligence, because in natural intelligence, you need to put our acknowledgement in a model, so you need to train a model with our acknowledgement, then you need to learn it, then you try to do more complex analysis that for human beings not possible, but you need to get this data and looking back by manual creation, whatever. So do not believe all the answer for ChatGPT or any of this GPT model, because there is a lot of mistake. Just one more final example. It's a little complicated, but I promise that there will be more step by step. So in this case, it was application plants. We tried to identify long no-code RNA is a machine learning. So I explain a little bit what's machine learning and what's no-code RNA. So what's no-code RNA? By definition, you have the central dogma of molecular biology. That's DNA. You have the gene. In your left, the transcription and the translation. So you have the protein. This is traditional. But in the last 30 years, you have the transcription of RNA, but we do not have the translation, and they have function. This is called no-code RNA. If you remember, last month, you have a Nobel Prize for microRNA, the small no-code RNA. So the small no-code RNA is considered up to 200, 300 nucleotide. And greater than that is long no-code RNA. So in our case, you talk a molecule more than 200 or 300 nucleotide size RNA. So that's the idea. So just to give an example what the biological problems, there is several classes. We are focused in bigger one. Why you have done that? This is a later on paper, but here is very clear that up to 2016, there is no tool for plants. All machine learning, prediction, whatever tools was focused more in human or mammals. And the question, of course, there is some, at that time I had some collaborator, and the question, okay, let's apply for plants. So we start to build it together in our department, of course, with some biologists for genetics. Let's do a machine learning for plants, apply for long no-code RNA. What's machine learning? In this case, was supervised machine learning. So basically, you have your label of your data. In this case, you know the fruit. So have apple or banana or whatever. And then you train your model with this label. So you're training what is apple, you're training what is banana. So the models you learn about that. This is supervised machine learning. So you're training the models of what is long no-code RNA, what's not long no-code RNA. Tatiana was my master student that have done this work. So basically, we start with two data. In this case, it's a binary classification. So the data set, a positive data set of long no-code RNA, and the negative was messenger RNA, the protein. And the idea is to say yes or not. After that, you have done some computational feature selection. You start with 5,000 features. Could be, for example, the color of your eyes, the size of your heads, and so on. These are features from sequence. And then you compare with 10 algorithms to get the best one. So the idea is the machine will learn, of course, and then see, okay, this data is a banana or apple, and so on. In this case, if it's a long or not no-code RNA. We start with, as I said, more than 5,000 features, and you get the best 16 features. So reduce the dimensionality of the problems and keep the same balance of performance. That's the idea. To show the same result in the view way, fancy figure, the top of the figure here is the raw data with no analysis, and the bottom we try to split between two groups. Of course, it's not perfect, but to minimize the error of to do this classification. Then you put like a line to split in these two groups. That's the idea, and you minimize the error. That's to summarize the machine learning idea. This was not only myself, but a lot of group, and you have postponed a collaboration with Germany that continues to do compare between human and the plants. So just for this slide for the student, what kind of knowledge was needed to do this work? So of course the biological acknowledgement about the plants, the long no-code RNA, etc. The data set preprocessing, more or less database, or data management, and programming. And artificial intelligence, in this case, machine learning. We're not talking about deep learning, was machine learning. So just to give you the basic level, what kind of knowledge was needed if I want to do that. What I want to show here, again, what you were interested to do is to building cutting-edge AI technique together with the biological data, because at the end you want to translate it in solution. That's, for example, in a tool, or in a database, or in a data set, etc. To finish my presentation, what in the future, this is my view. First, I think you are here about a lot of generative AI, and most of, for example, ChatGPT. You could make an interaction access for the model, and they give you answer, build the things in image, video, and so on. So this was the first comment about the tendency was 2022. By the Garden Group. This very beautiful woman does not exist, and if you go to this website, this person does not exist, and to reload, they will create a lot of face of people, kids, human, any type of people, and all is perfect, and all created by this generative AI. And of course, now I could create like I want a diet way they're surfing in the sea. And could create now image, you have Leonardo AI, Mind Juno, etc. You have a video. Now you have a lot of technology, not only by text, but video, etc. So now the creativity is up to us. So you call this kind of large language models that at the end it was initially focused for text, but now there is a lot of application. This model, the transformation, I think the changing point of this technology was 2017, was because our attention is all you need. That's focal, how you could get what you have previous, but how you could focus to create some relationship. Basically, to translate this model in more simple way, the idea that this model they will learn between relation, between the words. For example, if I get all the library, Wikipedia, whatever, I turn the model and see, okay, missing which are the relation, option, and so on. So if I type, oh, what's the next word if I have, we love, blah, blah, blah. We love you so much, etc., etc. Because he learns this relationship between words. And of course, what you could do now, you could apply for bioscience, life science, and so on. So basically the model, there is no limitation how this relationship between words happen. That was the change point. Of course, there is no free beer. Each of these aspects, chemistry, mass spec, protein, nanobodies, antibodies, is different problems, is different model need to look in, different new chain. Of course, I could train, Of course, I could look in and say, okay, I train like NVIDIA, train with all nucleotide sequence you have, but then you have different application because you know nucleotide sequence is one level, then you have a codon, you have protein, etc., etc. So depends on the problem here, maybe you need to create this different problem you need to focus. And there is no free beer because first the computational power is needed. For example, some models take a month to run. And then, of course, when you run your ChatGPT or any question, you are using energy and you're using carbon, so you need to be careful. I like to say people from computer science, it's very easy to click one in the bottom and put a model to run, but you forget to take time and energy and cost our plans. So let's think more. Let's think more about what you want to do, how you want to do, what kind of data, etc., etc. Then you put it to run and run and run. In my personal opinion, I don't believe that the artificial intelligence will replace the people. Let's just give a quick example. There is a very famous scientist and medical in Brazil, and he was a young doctor in a clinical, and there is a patient that arrived and they have done all the trial initial checkup, temperature, whatever, and everything is okay, but the guy is not feeling well. And they said, okay, everything okay, go, you could go. Before the guy stand up and go, his professor come and look at the guy and said, go right now to my room. In 10 minutes he have a heart attack. And they go to the professor and said, oh, I have done all the procedures, everything is okay. Why you know experience? 20 years of experience looking people die. They are normal, they stand up, they go and die. Then you saw some behavior. That's maybe the machine learning, they don't know because all the numbers, electrical, etc. is perfect for a model. But this is human sense. That's why, in my opinion, I don't think, of course, artificial will help a lot, but do not replace the human. So it's kind of help you to give you, oh, this is X-ray is 6% of benign cancer, whatever, etc. And the doctor have more information. It's really important to talk about FAIR principle, so we need to make it findable, reusable, available, all the data. Otherwise, if I'm from computer science, I want to do like drug discovery, DNA prediction, therapy, whatever. The first question I will ask you, okay, what is the data? What type of the data? If I do not have the data, so it's important to have access for that in a very user-friendly way. There is this paper in 2019, it's very interesting. They said, stop using deep learning, please, because it's a black box. I can't explain this model. So it's work, but how it's work, why it's work, what features they using. So people are start to say, okay, if you can't explain your model, why I use it? Although it's very fancy. Then it appears this new area that's called Explain AI, and now people talk about the interpretable AI. I need to understand my model, to explain how it's work. So now I need to explain how I build the house, not only build the house, not doing the black box. This one example is not my work, it's another from literature. What they have done here, this example, you have the image of a bird. So if you close to red, is the part of the model they use it to recognize that is a bird. Is closest to blue is the part of the image they using to do not recognize that's a bird. So they try to do this way to interpretable AI. What I have done with other professors and undergraduate students, you get a pneumonia X-ray image and try to see which are the parts more responsible for the classification, yes or not, this based on the image, is a pneumonia or not. So you try to, using this technique, to explain that. And of course, not only X-ray, but now you are fancy, you have UK Biobank project Genome English that you have a bunch of data and diverse of data, not genetics, but electrical, blood, brain issues, etc., etc. So it's so massive data that needs people and powerful to looking for that. Just a couple of, 1 year ago, when I start, I get this. So you have the mark of 500,000 genomes sequence. So you have a lot of data you could compare and see how it's going on in disease and so on. This is a bad way to use artificial intelligence, so I use the machine to do my homework. This is not good. What's a good example of how to use, for example, just have done yesterday, I asked for the ChatGPT, give me the title of my talk. Now you suggest another top 5 talk. Then you give me other options. Okay, I use my talk, I create my title, and I ask you if you could give me different ideas. This is a better way. Instead to give all the creativity process for the machine, let's use it to improve or polish what you have done. This is my opinion, what's much better way to use it. So just to summarize the key points, so I bring some example of bioinformatics, biological, and artificial intelligence. Of course, I strongly believe in AI in biology, and of course, we use the data, you apply artificial intelligence, but at the end, you want to extract insights to the decision making. These are the people in Brazil that I have done collaboration in the last 14 years, and this is my new lab and the group, some people of the group of the Rosalind Frank Institute. And Mark Basham, you'll talk with you in the afternoon, you'll talk more a little bit, other things you very fancy do, very nice in the Franklin. And of course, this is for the students. If you want to come to the dark side of biological data science, you could take a picture if you want of this slide. It's most of the reference of books. Old school, I like print books, sorry. I still like book. So if you want to go to this side, this kind of reference, I really recommend. There is more introduction for more technical one. And just to more marketing, you have next year in May, I organize this Rainbow course. It's more scientist, postdoc, master student, early career. But you'll be here in Didcot, the Rosalind Frank Institute, this transpositionamento in the area of data science in May. We still have time up to now in December to do the application, be free. And just to finish it, let's message it. Remember about that? So it's computer time, and then, and of course, any option that you lose, but at the end, the computer wins. Of course, you found a different way to give the answer. That's why you call hallucination. The machine learning, or AI, you give the answer, you need the human to confirm if this makes sense or not. And of course, I don't believe that computer science or biology or medical is the best science, but in my opinion, collaboration is the best way to do science, because I'm from computer science, but I need to do collaboration to avoid to do mistake and do more powerful science. And of course, coffee is crucial. Without coffee, it's not possible, or tea, it's not possible to do our work. So I'll finish my talk with, I love this phrase from Einstein, that the definition of insanity is the repeat to do the same thing and expect different outcome. Thank you.
Does anyone have any questions to ask? So given the potential variance in biodata, specifically from when you're taking it from many different sources, how do you reconcile issues that may arise from introducing that uncertainty into machine learning models?
Good question. How many time do you have? In summary, when you have this data and you compare, you have metrics that you try to see how good is your model. accuracy, sensibility, precision, recall, that try to give you, if you try to find how good you are and something that's not what you want, how good you are also. And there is different statistical methods, but the general answer is that when you have positive, negative, in the case that you are showing you, the machine learning. But depends on the case, there is different approach you could do. But there is statistical methods support mathematics to confirm, to evaluate your model, and of course, to compare with the other from literature.
Any more questions? Can we have another round of applause, please, for Dr. Paschoal? Okay, because you've been sat there for quite a long time, we're just going to have a bit of a break, okay? So about 1:45 to have a break. There's food and stuff like that downstairs, toilets downstairs as well if you need it. Then it's the time for you guys to get involved, okay? So your posters are all at the back. The judges are going to be coming round at about 1:45 to discuss your research and see who's the best poster is for the day. So please do be up here to chat about your posters. Thank you.
really enjoyed looking at all of your posters during lunch, because they're really, really excellent examples of how to communicate the science that you're doing to somebody who isn't a specialist in that, who hasn't spent the last couple of months, or in our case, multiple years, looking at a particular thing in detail. So, I'm going to be adventurous and try and use the clicker here. There we go. So, who am I? My name's Mark Basham, and I'm a researcher. I'm Science Director for Artificial Intelligence and Informatics at the Rosalind Franklin Institute. That's very much my job title currently, but the things that I really enjoy nature. I spend a lot of time trying to make my life and my house as carbon neutral or carbon negative as possible. I drive an electric motorbike, which is very fun. I have an electric car. Spend a lot of time in woodlands. I'm Celtic, so I'm originally from Cornwall. I enjoy Celtic art because it's quite mathematical, and I'm terrible at normal art. And I enjoy now, it used to be a little bird up there, but I now enjoy the delights of blue sky and social media and trying to communicate the work that I do to the widest audience possible. And I work up there at the Rosalind Franklin Institute. Now, oh wow. Okay, so I wanted to highlight this. So I'm a first generation university student, which means that none of my direct family, nobody that I knew when I was growing up, had been to university. And so there was a whole bunch of stuff that I found incredibly confusing. And you may well know this in advance, but if you don't, be aware if you do go to university and you see this kind of stuff, it's the sort of thing that everyone is, or there are definitely going to be other people who are not familiar with it. But I just wanted to tell you a little bit about my history and how that makes up my current name. So, first of all, GCSEs, I studied the standard set of things. About the only thing that was sort of chosen there was that I did the separate sciences, so chemistry, biology and physics as separate entities. And I chose technical drawing as my art. I'm colourblind. I am incredibly poor at actual art, but technical drawing was something that I really enjoyed. I really enjoyed being able to do the detailed study of that. When I was going through my GCSEs, I really wanted to be a pilot in the RAF. So I was an air cadet, I really enjoyed flying, and that was my main dream. At GCSEs, I got a D in my English, which meant that I was no longer able to pursue that as a career option. It was kind of a bit of a setback for me, but actually during my A-levels, I resat my my GCSE English to get myself a C, which enabled me to sort of carry on my career in the future. So, minor setback, but it sort of changed the direction of what I was interested in. But it meant that I focused a little bit more on my A-levels. Now, my A-levels, I didn't think they were going to be that important, so I just chose things that I enjoyed. I did maths, physics, and design technology. And so through the process of that really led me to really understand and really be interested in engineering and robotics. And so at that point, I really wanted to go to university to study robotics, study cybernetics. At my A-levels, I wasn't great in school. At my A-levels, I got 2 C's and a D, and I needed 2 C's and a B. For me, I just did not get pure maths. Just didn't click for me. Mechanics, I absolutely loved. Pure, couldn't get my head around it. So I got a D in maths A-level. So I'd got an A in GCSE, done it multiple years early. It was my top subject. Absolutely didn't get it at A-level. So I rang up the university and I said to them, what other options are there? Have I still got in? And they were like, no, I'm sorry, you haven't. But we've got a really nice, interesting new course called Physics with Computer Science. You might be interested in that. And so I thought, okay, well, I may as well have a go at it, see what it's like. Apparently, a lot of the courses were similar. So I went and did that. And for me, university was basically the place where I could really excel. It was the place where I learnt in the best way possible. First year, we had a great course on maths. It brought everybody back up, but also it was for me where pure clicked, and I was able to really go well with that. And so essentially I went through my degree really enjoying that. And so you can see that's highlighted because that's essentially where my BSc Hons comes from. So this is just a general thing in your name that highlights that you've got a Bachelor of Science, and Hons means with a 2-1 or a first. That highlighted that I was able to then go on and do some interesting things, and I really enjoyed chaos theory. So this is really a computational mathematical problem. And so chaos theory was what led me to do my philosophical doctorate, which is a doctorate in sort of research, specifically on computational science, which is essentially why I'm a doctor and where the PhD, philosophical doctorate at the end of my name, comes from. And essentially, over the course then of the last sort of next 15 years or so, I spent a lot of effort doing a lot of interesting projects, working in cool places. And I've always been a member of the Institute of Physics, which is a sort of the national physical sort of association, National Physics Association. And I put all of the stuff that I'd done together and applied for a fellowship there, which now is what I'm a fellow of the Institute of Physics, which is the final bit there. So essentially, that kind of tracks where I went and the sort of choices that I made at university to get me to where I am now. And where is that? Well, that's the Rosalind Franklin Institute. So this is a sort of a reasonably new, it's about 5 years old now. The building itself is about 4 years old. Institute it's in Oxfordshire. So here we are on the map. So it's just south of Oxford. And it's based on the Rutherford Appleton Laboratory. So this is the UK's sort of one of the UK's national science infrastructures. And it's something you probably haven't necessarily heard of unless you've been explicitly directed to it. Essentially, where all the universities do research across the country, when they need to do research that's particularly big, so things like using particularly large-scale experiments, really big lasers, really big stuff, then they come to one of these central locations where there's all these central facilities. So here we have things like Diamond Light Source, which is the UK's National Particle Accelerator. We've got the ISIS Muon and Neutron Source. And let's just go to a slightly different angle. You can sort of see them across the background. And it's all sorts of scientific kit that you may want to use if you're at university and enables us to do stuff that's not plausible at a university, one university, sort of national scale science infrastructure and national scale science that we're interested in. So that's where it is, but what is the Rosalind Franklin Institute? Well, it's very much based off the namesake, which is Dr. Rosalind Franklin, and she's probably most famous. So she was the scientist who collected the data for Photo 51. So Photo 51 was the X-ray crystallography photo that basically initially sort of seeded the thoughts for the fact that the structure of DNA was a double helix. So so this is X-ray diffraction. You get a specific pattern which is based on the structure of the shape you're looking at, and a double helix gives this kind of pattern. And this was the work that was then taken with Watson and Crick and effectively got them the Nobel Prize. Unfortunately, at that point, Franklin had passed away. And also there's quite a lot of controversy around this about the fact that she wasn't credited enough when she was alive for the work that she did as an instrumental part of this, because it was by taking those physical science concepts, so X-ray crystallography, and bringing them to the biological sciences, to sort of DNA, that enabled these insights to come about. So hence the Rosalind Franklin Institute is very much based on bringing physical science concepts into the life sciences. So we work a lot on using all kinds of different technology like electron microscopes, mass spec imaging, all sorts of things to bring more and more information about cells, biology, organs, tissues that we're interested in. So when I got asked to come and present here, I was thinking, well, what am I going to talk about? And one of the things that I thought, I often get asked when I come and do so I do quite a lot of outreach. I work with schools quite a bit. And one of the things that I thought I was really interested in was I get asked this question a lot. What part of STEM do you work in? And I think, especially at school, a lot of this is broken up. One of the things I loved about your posters was so many of them had blended elements of STEM together, because it's really the sort of stuff that we do. Because what do I work in? Well, I work in all of it. And I really like the new acronym STEAM, which adds the A in there for arts, because actually a lot of the work I do is design and thinking about how people are going to use the things that I create. I'm a software developer, so my background is writing software. And understanding how people are going to use that software is critical so that I can try and make it as effective as possible. I could write the best piece of software for solving a physics problem or a maths problem ever, but if it was horrible to use, no one would use it. And so making those artistic elements as well are critically important. And so, yeah, I've worked on large pieces of equipment. So this is the inside of a particle accelerator. I've done huge amounts of work with large amounts of compute and infrastructure like that, building small-scale equipment, all of these things. And all of them require me to use some of the science that I've learned to understand why we're doing the problem, the maths to basically as the workhorse for how all this stuff works, engineering and technology to actually build and put these things together. So it kind of hit me, though, that actually when we talk about science, sometimes we can talk about purely just the bits of science we're doing and not necessarily think about some of the technologies that make that, kind of enable that. And I really like the technologies. I think they're the bit that, for me, is really exciting. And so I think I started a lot of the work that I did in public engagement around talking about technology and science. And so I was trying to really think about how I could use technology to sort of tell the story that I was interested in. And actually, one of the first things, this was an amazing exhibit that we went and did up at the National Eisteddfod in Wales. And one of the things we took with us was this screen. So this was probably about 15 years ago, and this was just at the dawn of when 3D screens were possible. And all of a sudden, so this just looks blurry in this image, but this was actually a 3D screen. You pop the glasses on, and these proteins that we're seeing at the left and top, and a 3D model, so a 3D X-ray tomography image of a Raspberry Pi at the bottom right. And all of a sudden, these things could pop out, and people were immediately engaged by it, and they were immediately sort of excited by the fact that there was this cool technology they could play with, and potentially they could even have at home, because although they were a bit expensive, you could buy one of these. You could just go out and grab a TV, which was nice. And it kind of really hit me that this was using these technology and different mechanisms was a really great way of interacting with everybody, interacting with the public. So after that, I was sort of looking out then for more opportunities to engage with people. And I got, I sort of signed up to do this amazing project called I'm a Scientist, Get Me Out of Here, which if you're a teacher, I would thoroughly recommend looking up. Essentially, when I did it a while back, it was very specific, but now it's very, very general. You can speak to, it allows you to get your class to speak to a group of scientists on pretty much any topic. And you can just sort of, it's an interactive chat. It's just a text-based chat, but the fact is you get to ask any questions you like. You get asked questions all around what your favourite films are, what you like to eat, all sorts of stuff like this. But in that, one of the things, when I did it at least, you had to choose, the idea was that the students who talked to you got to choose which of the 5 scientists that were in this sort of room together would win a £500 prize. So it would work over, similar to I'm a Celebrity, Get Me Out of Here, you would eventually get voted off until one person remained who won the £500 prize, and that was what the students got to choose. And so you had to choose what you were going to do with it, and it had to be spent on public engagement. And so at this point, I was very much used to looking at all the kit we have at Diamond. So I used to work at Diamond Light Source, and we had all this amazing kit. And when I took people to see it, it was always really impressive, but I could never take that with me to an exhibit anywhere. And so what I thought was, well, £500 is just about enough to buy one of my favourite toys, which is Lego. And so I was in the situation where I was like, well, I could spend £500, I could buy some Lego Mindstorms, and maybe I could build one of these beamlines out of Lego. And then I could take that with me because it would be quite small and portable. That might be quite fun. And so, but I had to win. This is very, very important. And this was the period of my time where, as a physicist, it saddened me a lot, but I learnt to really, really hate the standard model of elementary particles, explicitly the neutrinos across the bottom. And the reason I hate them so much was, as I said, it was a 2-week period. And in the middle of that week, in the weekend, I was doing really well, I'd like to highlight. I was definitely going for first in this competition. I was in the big facilities meeting, so there was someone from lasers, there was someone from CERN, and there were a couple of people who were studying elemental particles at really deep mine sort of based places. And unfortunately, in the middle of all of this, one of the experiments that one of those people worked for mistakenly observed neutrinos travelling faster than the speed of light. And this hit the media everywhere. So every single news channel you went on, all the newspapers were filled with this. And it turned out that they hadn't plugged one of the cables into the back of the instrument correctly. So there was and they suspected this was the problem, but for a short period of time, this was all over the news. And so I came third in that competition. I was very, very disappointed because they got all the questions for the second week. And I was very sad. Luckily, one of my colleagues, who was basically part of the communications team, said, actually, it was quite a nice idea. Why don't you go ahead and build one anyway? And so I built the Lego beamline, which was a diffraction experiment. So the idea, you can see there's like a laser pen, which is the shiny tube. It shines through a diffraction grating, just the same as you do a normal experiment. But the cool technology was the fact that the arm would swing round and it would draw out a plot of the data that you'd collected, so you could work out what the spacing on your diffraction grating was. And it turns out that this was really quite popular. We went to lots of things, and there were various different versions of it. So this was the first one. I haven't got a video of this, but I have a video of the second version. Sorry, this is incredibly difficult to film while interactively doing it, so I do apologise. But you start it going, you shine the laser pen, this arm spins around and it starts measuring the spots of light. And when it measures the spots of light, it drives the pen up and down to plot that on a graph so that you can then essentially take this off and you can see where those were and you can measure it. It's essentially the technology that we use all the time in these big synchrotrons where we've got X-rays. You can't go into the room with them, so you have to remotely collect all the data. And effectively, this is exactly the sort of thing we do, but on a slightly larger scale, with slightly higher precision. However, with LEGO, I could get precision of about 500 nanometers without too much issue. Yeah, big synchrotron, big X-ray system, you can get a lot higher precision. And of course, it carried on going, so we've got other versions as well. So this is the third version when they changed to the EV3. But the idea of like being able to represent that sort of technology was great, but in a way it wasn't really the area I was working in. I did quite a lot of work with the computational side, and there wasn't too many ways of interacting with that, and I was really interested in how that would work. So this is one of the server rooms that we use at the Franklin now, but also at Diamond. It's based on this huge site. We've got huge amounts of compute there because we process ridiculous quantities of data. The Rosalind Franklin Institute, for example, has just passed a petabyte of data, and we've been collecting there for about 2 or 3 years. We're really quickly wrapping up to collect 2 petabytes. So a petabyte is 1,000 terabytes of data, and a terabyte is 1,000 gigabytes of data. So I guess a Blu-ray disc or a movie is probably about, I don't know, maybe 10 gigabytes of data, that sort of area. So it's huge amounts of data, huge amounts of sort of content that we need to look through. And to do that, we use a lot of compute. We use a lot of these really sort of high-performance computing. But again, it's really difficult to show that and to represent that to people because this just looks like magic, right? Nobody, unless you actually work in the industry, you've no idea that this is actually just a bunch of computers. But again, there's commodity stuff that happens that we can use. So hopefully most of you recognize this. This is a Raspberry Pi. So this is like a £30 computer that you can get for schools. It can do all kinds of really useful things, but it is literally just a computer. And if it's a computer, we can build it into a cluster to do all the high-performance computing that we need to do. And so you can build yourself a little beautiful Raspberry Pi cluster. And this one we used to do some chaos calculations because I like chaos. So we used it to generate the Mandelbrot set as quickly as we could. And it would split up the task across all the computers, and it would do a little bit of the task on each of those computers and then bring it together at the end. And this is exactly how all of the high-performance computing pretty much across the world works. So when you're using your latest ChatGPT model that's been trained on however many billion compute nodes around the world, this is essentially what it's doing. It's a relatively simple process. And by bringing it down and just looking at Raspberry Pis, it kind of makes it a little bit more accessible and allows you to communicate it in a bit more easy way. Of course, if you want to then look at some other things as well, so this is another beautiful Raspberry Pi-based cluster. So this is actually creating an object store. Now, an object store sounds a little bit strange, but effectively, if any of you have used Google Cloud or Dropbox or OneDrive, all of these store your data, store your files, your photos, your images, somewhere remotely on the cloud. And behind all of that is a compute system that's remarkably similar to this one, with loads of different computers, each of them storing a little bit of information. And the idea is that each of these little computers here, you can actually just turn them off. And as long as you don't turn off too many of them, everything keeps going. That's why, generally, your Google Drive is always there, your Dropbox is always there. You don't have to, there's not downtime or anything like this. And these sorts of systems work really well at doing that. Okay, so we've got some nice compute systems, but really one of the areas that I was interested in was basically collection of data. So when you're doing a science experiment, often when you guys are doing it, and actually you've got some really amazing examples of doing some great experiments where you're collecting data. Now, in modern big scientific facilities, we collect all of that data electronically. It's very, very important that we collect it all as quickly as possible. And that we do we change the variables and the parameters of those scans as effectively and as precisely as possible. And so to do that, we use various different pieces of software. One of them is called GDA, which stands for Generic Data Acquisition. And it's all about how do you set up an experiment to run on a computer that talks to all sorts of bits of compute and hardware, motors, robots, and all sorts of things to do the experiment for you so that you can sort of do that in the best and most repeatable way. And so, of course, we wanted to see if we could run this on a Raspberry Pi. Why not? You can run most things on it. And so what we did was we created a system. This is kind of a view from the Raspberry Pi camera. And you can see there's a little turntable there. And the idea was that we could put a model on the turntable and we could rotate that model and collect lots of images of it. And that would produce us a 3D model of that, of whatever we put on the turntable. So this is kind of analogous to a technique that we use a lot at the synchrotron, which is called X-ray tomography. So if you've ever had a CAT scan at hospital, so it's a computer-assisted tomography scan, the idea is you lay in a tube or put an arm in a tube, and an X-ray source goes around you and a detector, and they can reconstruct a 3D representation of you to see your bones, all of the muscles inside your body. We do the same thing at the synchrotron, but it's very hard to rotate a 500-meter diameter particle accelerator. So what we do is we spin the sample and we image it with a single beam. And so in this case, we chose Lego as the primary choice. So we chose a little Lego figure, we rotated it, we collected a bunch of images, and then we could use some processing software to create a 3D model of that. And we were quite happy with this. It was a really nice summer student project that we did. It was really useful because it enabled us to actually get our software out to some groups who really wanted to use it but didn't have the kit locally. So we were able to just post them a Raspberry Pi with everything set up on it and an Arduino and some little motors so they could test things out. But it also meant we were able to do this quite nicely. And it was kind of good, but it was a little bit tricky to use and it was a little bit difficult to interact with. And so, kind of as a technology example, it wasn't great. So we were a bit stuck as to what to do with it. But then we came up with a different project. And this was my first introduction to the idea of citizen science. And citizen science, for me, has really transformed the way that I do the research that I do. And it's sort of come about from this sort of exploration that we did. So Project M was all about this idea of, they wanted to do an exploration of calcium carbonate and the various different sort of additives that you can add to it to see how that changes the structures that you get from it. And the reason it's called Project M is because M is 1,000 in Roman numerals, and so they wanted to do this with 1,000 samples, so they had enough replicas. Now, what was interesting about this was that to do this, and so I think calcium carbonate, I think you create that in A-level chemistry, and so what they were interested in was could they connect with 100 schools around the country, each of which was doing that experiment, and get them to make the samples for them. Because actually for us to make the samples where I was working would have taken too long. It would have taken a researcher maybe a year or so to go through all the processes to do this, whereas we could actually kind of crowdsource it and outsource it to citizen scientists really effectively. And so essentially this group of people, the people on the left, were basically planning this project, and they were using this beamline on the right-hand side, and you can see there's like a big blue robot arm, and that robot arm can change the sample very, very quickly. And this enabled us to go through like 1,000 samples in a single day. So it would load the sample, it would collect an X-ray diffraction pattern, very similar to what Rosalind Franklin collected, but that would tell us the structure of the calcium carbonate that had been created. And then, effectively, it would then put it back, move on to the next one, and keep going. And so the project was running. We had students all over the country basically creating the chemicals that we were, or basically doing the mixing, preparing the samples, and packing them into these sort of little gold, I think they're actually copper capillary tubes, which are where we put the sample to enable us to scan it and to load it onto the robot. And so this is, I think, all of the 1,000 samples all ready to go. And then the fateful day came, and we wanted to load it all up onto the robot and see how it would go. And so I was involved in none of this until this very last point where they were like, oh, but wouldn't it be great if we could see how, if the students could actually see when their scans were being done and when things were happening. And so, as I say at the beginning, at the time, I was interested in Twitter. And so what we did was we created a nice little Twitter bot that basically for every single scan our system did, it tweeted the result of the data collection that had happened. It tweeted a couple of pictures that were taken from webcams all around the thing. And if we knew it, it included the tag of the school that was involved or what was going on. And it just enabled us to really quite, interestingly, gain some engagement and try and actually represent what we were doing. And it turned out, this was the interesting bit about this, was it turned out it was relatively easy to use the Twitter API to do this. I think we got banned about 2 times during this process for putting too many tweets out in a short succession. But over the course of the day, we tweeted the majority of the results, and everybody got to see what was happening as part of the experiment. And as I say, it allowed us to really see how you could use Twitter as a way of potentially communicating results from experiments back out to to people. And this really made us start thinking, because we thought, well, hang on, actually, if this is a great way of communicating results out, maybe we could use it with our Lego beamline, with our Raspberry Pi beamline. And so we did a little bit of work. We tried to upgrade our Raspberry Pi beamline. We sort of were inspired a little bit, and we put a robot arm in there to allow us to change the samples. But it's still just the Raspberry Pi, the camera, the detector, and a few bits of extra kit to make it work for the sample changing. But now we were sort of armed with this idea of, well, wouldn't it be great to control it? Could we not do the entire thing and control it all through Twitter? So now, on Twitter, there's it unfortunately no longer works, which is very sad. It's been turned off. But there was a system called Diamond Raspberry Pi, and what you would do would tweet it a message saying, please scan sample 3. At which point, what you get is you'd get a series of videos. You'd get a video like this one showing it moving the sample and putting it into place. You'd get a video of it collecting all the data, and then it would reconstruct it, and you'd get a video of that kind of model at the end. And it suddenly meant that this thing that was kind of not very easy for people to engage with or work with, through a little bit of extra work in terms of using Twitter to do this, we were able to suddenly make this much more engaging. And it was great for an awful lot of different projects that we wanted to do, like careers fairs and stuff like this. So there's quite a lot of really sort of interesting things going on with there in terms of how we wanted that all to work. Now, at this point, there was an awful lot of stuff that I was really thinking was going sort of really interestingly. We had lots of really engaging and interactive things, but one of the areas that I was super interested in was whether there was any way that we could do sort of engagement and talk with people on a broader scale. So where we could sort of say, well, actually in an unfacilitated way. So all of these things required me or someone to be there and actively talking about it, and it was great for that. But wouldn't it be nice if we could just encourage people and talk about stuff that actually is really important in science? I was having a conversation with a few people over lunch about some of the really important parts of science, which is very, very different from what you do in school, which is all around failure. So when you're going through school and when you're going through your degree even, what you're trying to do is succeed, and you're trying to get the correct answer. You're trying to solve the problem in the right way, and you're trying to get the correct answer out. When you hit sort of research science, all of a sudden, probably 95, if not 99% of what you do doesn't work. It fails. You don't get the answer you want. And it's a really big hit for people because people are used to succeeding, right? Because that's what you've learned. And suddenly hitting failure all the time it can be a really difficult step to overcome. And so we really wanted to try and communicate a little bit more about how research science, there was a lot of failure involved. It was something that people should be happy about and be aware of, that you're not going to be able to succeed all the time in these problems. And so, as I say, at the time, I was working at this big shiny building here, Diamond, which I've talked about a little bit, which is this large particle accelerator. And my current building is just up behind it, which is the Rosalind Franklin Institute. One of the reasons why I moved, one of the reasons why these science parks are great, because loads and loads of people change job within this science park, but actually you don't need to move house or anything. There's loads of nice, easy ways for you to jump around without having to upturn your whole life to move. But so we thought, well, what would be a nice way of doing this? And we decided, well, let's have a look at creating Diamond the game. So we wanted to create a completely sort of autonomous and something that somebody could just go and do on their own. And we thought, well, actually games are a great way of doing this, because generally you don't have someone teaching you how to play a game. You generally buy a game, you read the rules, and you then go and play it. We also thought, well, hang on a minute. Diamond itself pretty much looks like the board already, so we're on to a winner. We can just sort of take that across and make use of this in a sensible way. Now, luckily, the people I was working with, so Claire was one of the scientists who worked on Project M, and Matthew Dunstan was a scientist who'd actually moved across into doing game development professionally. So we had lots of people who we could talk to about how to get the best out of this game and make it as exciting as possible. Because it really was important to make it fun to play so that people would be interested in it, but also that it sort of got across the points we were interested in. And so the messages that we wanted to get across was, well, we talked to secondary staff, and actually curriculum-wise, careers was a really important point, because there's so many careers that are available. So that site that I showed you a second ago, there are about, I think there's now 7,000 people working on that site. About 2,500 of them have PhDs, and the rest are technicians and other support staff, like, you know, people with law degrees or financial degrees who basically make everything actually work. And so there's this huge array of different careers that are available that aren't necessarily the ones that you immediately see. So we wanted to bring that to life a little bit and show some of the different careers that we're about. We also wanted to highlight, I mean, it was Diamond and we were doing this deliberately, we wanted to sort of highlight the breadth of different science and stuff that was available there. And we also, as I said, had previously, wanted to talk about failure and collaboration and how actually current science, very, very little current science is done by one person. It's almost, most of the low-hanging fruit that you could do on your own has been done. Most of the science that currently happens is done by bigger teams, bigger and bigger teams, and it's really important that the collaboration, the communication elements are all there. So we also talked a little bit about actually trying to make the reach as big as possible. So we deliberately tried to design everything. We worked really closely with the graphic designer to make everything high contrast. So if you have visual impairment, it was going to be easier to engage with it. For me, all the icons were made so that you were colorblind friendly, so that you could really interact in a nice way. And we spent a lot of time actually trying to think about this to make sure it was as accessible as possible. We spent quite a lot of time playtesting it. So I spent a lot of lunch times playing the game with loads of people from around Diamond, all about trying to make it fast and, to iterate it quickly, but predominantly to make it fun. Because, as I say, being fun was really, really important. And then one of the key things, actually, which I really like about some of the public engagement stuff was trying to bring back the sort of scientific element of it to a certain extent and was evaluating it, trying to get all of that information back to us so that we could understand whether it had done the things we wanted it to do, whether it had potentially engaged people in science, made people more aware of different things. And so after we'd done a load of initial tests with a load of schools, we got some information back, and it basically sort of said that actually, you know, I like science. Pretty much before and after they were roughly the same. But actually what we had done was that people had thought about would they like to be a scientist or engineer was up massively, because hopefully not because they'd suddenly gone, actually science is really cool, because we know they didn't like science anymore afterwards, but hopefully possibly just because they saw that there were a few more careers in there that they weren't aware of before and that they thought might be interesting. And also, we got a big uptake in the number of people who saw science in their day-to-day lives, because we started talking about projects that were not necessarily the big astronomy or big particle stuff, but also just all the stuff that you have on a day-to-day basis, like making good glass for insulation, or making new materials, or making drugs and things like this. And lots of people had fun, and that was the most important bit for us. So then we thought about how do we sort of scale this up and make it work. We sent it round to 100 schools, so we sent 100 schools around the UK, a box of this so that they could play. We got some really good feedback, and it was really nice. And when this all happened, it was just before 2020, and so 2020 was very, very challenging. I'm not going to go into too much detail, but obviously the pandemic hit, and there were an awful lot of people who were suddenly stuck at home. Basically, everybody became an educator because you had to deal with this. And we thought, yeah, we were also stuck at home. We weren't able to get into the lab, and we weren't able to do much stuff. And we thought, well, maybe we could change what we were thinking of doing. You know, this box set was quite difficult to send out. We only had a few copies of it that we weren't able to send to everyone. But what we realized was that we had a bit of spare time, and we could potentially redesign it to make it into a print and play system. So the idea here is that this is just something you can print out on your local printer, and then you can essentially just sort of have a go at this yourself. What we realized was obviously all that high contrast stuff we'd done in the first case was really going to sort of be bad here, because all of a sudden everything was incredibly dark and you'd used a lot of people's ink. So we deliberately sort of changed everything round. We tried to scale it down a little bit and make it more easy to print out on a smaller number of pages. But what was really nice about it was that suddenly this was something completely digital that we could send out to everybody. And so this challenge became a real opportunity, and sort of to date we currently have about 10,000 players in about 30 countries across the world. It's very much, we built it in English, so it's very much sort of dominated by the English-speaking countries, but there's a few ones that go outside, which is really nice. And essentially it just enabled more and more people to sort of access this and hopefully gain some useful information, if nothing else, just sort of have a bit of a chance to play around with the game. What also became really interesting was that actually we learned so much through this process that we really wanted to talk about it. And so we published it specifically. We wrote a paper all about how we did this, which effectively was there so that the public, the public engagement community could sort of take on anything. If there's anything that they wanted to take forward or do use, they could make use of that. So, how does any of this help science? And I kind of hinted at it a bit with the Project M thing, in terms of by using the public, we were able to actually do a lot more samples than we're able to do. Now, my work is all in artificial intelligence, and we do an awful lot of image processing, and computers are really bad at image processing. So, over the last probably 5 to 10 years, we've got better at doing basic stuff. So, when you take a photo on your phone, it will often prompt you that that's maybe food. If you take a photo of pretty much anything circular, it will probably prompt you that it's food, because it thinks it's a plate and that's what you want to do. There are cats and dogs and stuff it can identify pretty well, because there are thousands and thousands, if not millions, of images of cats and dogs on the internet that we can use to train our systems. But when we get to more complicated things like biological systems, there isn't all that training data. And it makes it very, very hard for us to teach artificial intelligence systems how to do stuff. And generally, even if they do it, they do it quite badly. However, people are quite good at doing this kind of work. So we now do an awful lot of work with a group called Zooniverse, which comes from the astronomy community, which is all about getting the community and getting citizen scientists to basically do tasks that computers find incredibly difficult, like image processing. So what we do is we take our biological imaging data, so maybe these high-resolution images of cells or of neurons or of tissues, organs, and we basically break those up into smaller sections that the tasks can be done more easily by citizen scientists. We then put it into our Zooniverse platform, and we get people to basically annotate this for us. And then we essentially take that back, and we then use our skills to bring all that data together, potentially retrain new artificial intelligence models to understand and to essentially become better at doing what they're doing. And then we apply it to more data, and then we cycle back around to the beginning. We hand that back to the citizen scientists. They help us by tidying up any of the mistakes and things like this. And essentially, by doing this, we can do what would take a single researcher years, if not decades, to do, because the technology is getting so good now, we can collect so much data, that it is literally years, if not decades, to keep up with that in terms of researcher effort, annotating and dealing with these imaging data. We can do that all through citizen scientists, and we can do a load of things very, very quickly. So the project that we actually have open at the moment is called Science Scribbler Synapse Safari, which is an excellent illustration. And so we have, the idea is that we're looking for different, so we're actually looking for mitochondria inside cells, specifically inside synapses. And with the system that we've got, has automatically identified some of them, but a few of them are incorrect. So they're in the wrong sorts of places, and we need people to go and correct those, tell us where we've got it wrong so that we can retrain our models and get it better. And the reason we're interested in this is because of all sorts of neurological diseases, and we're trying to understand exactly how synapses grow and form during a sort of early life. So these are really excellent ways of doing this. Now, previous work that we've done on this platform has actually been all around basically using viruses and identifying where viruses are in cells. So we looked at reovirus and rotavirus. And there's a nice little pack that you can get. So if anyone's interested, please come and talk to me afterwards. But there's a pack you can get for schools, which basically has a whole bunch of, it's got sort of 4 lesson plans in it, all around sort of the biology and computer science curriculums. And it explains how we use sort of these algorithms to essentially to explore this data and to understand the results of it and how we sort of visualize it. And it's quite a nice interactive sort of process. So that's pretty much all my time. I've put everything together on the end, all the way through from I'm a scientist, which I thoroughly recommend as just a great way of communicating with scientists, through Synapse Safari and Science Scribbler. If you're interested in actively helping us out with the research we are currently doing on neurological disorder. Virus Factory in schools, if you think it might be interesting for our outreach that's more to the teachers. And then Diamond the Game, if you fancy playing a game that's based around being a scientist at a particle accelerator and trying to do some cool science and beat your peers and do the best science. But with that, that's pretty much all my time. So thank you very much for your attention. And if you have any questions, please feel free to ask.
Are there any questions?
Where did you produce your board game and other materials?
So there's two places. So the first one we did, unfortunately it doesn't exist now, but there were places which were print on demand. So we used a place called Ivory Games that still does cards, but it doesn't do the full board games, and we built the prototypes with that. But then we went so we did a mass production run. We built 1,500 of them, and we used a company in Poland called Media Gold for that. But it turns out that shipping 1,500 games is harder than you probably expect it to be. There's many pallets, and it was confusing.
Any more questions before we move on? No. Can we have another round of applause, please, for Dr. Basham?
Oh, yeah, no, of course.
Just to give us a little bit of time.
Thank you. I'll utilise that as well. Sorry. I'll come down. So I think you just leave that one alone. He can mute you.
Thinking about science communication. There we go.
I just clipped it in my pocket.
Yeah, I think that's probably where it is. Something to be done on the clock. There we go. Okay. Sorry? Okay, we good? Right. Okay, so hello everyone. My name is James. I work in science communication. And so my background actually is I was a research scientist for a while, and I wasn't very good at it, frankly. I found it really quite dull doing the same problem over and over again every day. So I moved more into science journalism, worked with the BBC, and now I work mainly with research institutions thinking about ways that they can present their science to the public, also to policymakers. It's very important that politicians understand science, and through the media. So can I just ask you all first, who here ever accesses any kind of science media, podcast, TV show? Who does any of those things? Almost all of you. What do you watch? What do you listen to? BBC Bite-Sides. SciShow. I'm hearing some new ones here I haven't heard before. Any others? Right, and how do you know that what you're hearing on those shows is true? You don't. And that's the biggest problem that we have in science at the moment. We have never lived in an age where there has been more mistruth, misleading, conspiracy theory out and about on the internet about scientific subjects. And that's a big, big challenge for science, and it's a big, big challenge for society. So I'm going to talk about science communication in a slightly different way to what Mark's been talking about. He's talking about, as a scientist, how he works with public engagement. I thought that was a terrific example of what you can do to try to bring about change. But actually, we have a much, much bigger problem with science, which is that we don't communicate it very well. You're exceptional in that you're interested in science. And almost all of you are accessing science media. But actually, most people aren't that interested in science. And that's a real problem, because unless we can get people to understand what science is about, we're going to suffer more and more difficulties in trying to tackle some of the great problems that are facing society today. Now, I work at an institute called Rothamsted, which is actually the oldest agricultural research institution in the world. And the reason we're trying to do the science that we do is that our food system is in a complete and utter mess. It is probably the single most destructive thing that we do on Earth is producing food. Clearing away biodiversity, draining swamps, clearing away forests, creating land for us to have the food that we want. It is a massive, massive problem. We need to radically change the way that we farm, but food is a very important cultural feature of what we do. We're very emotionally tied to what we eat. So actually presenting the science about how we can change our food system becomes extremely difficult when people are very attached to what they might term natural food. And I'll show you an example of that as we go through. But we need to communicate the science that we do. A lot of the science that gets published, it goes into a journal, it sits on the shelf, nobody looks at it. So taking science that extra step, just as Mark's been describing, is absolutely vital in order for us to get people on board in supporting science. And a lot of science is publicly funded, and we do need support. So just in the area I work with, environmental scientists, very few people feel that they understand what's going on. They've heard of climate change, they've heard of deforestation, but they're really not sure what's going on. They listen to scientists a bit. Scientists, there's a thing called the Edelman Trust Survey that comes out every year. Scientists are normally near the top, estate agents and politicians down at the bottom, not trusted terribly at all. Scientists do have a lot of credibility, but that is declining. People are trusting scientists less and less. And we have a problem in the policy arena as well. So this is from Parliament 2 parliaments ago. I haven't got the figures for the current Parliament. I would suspect it's not that much different. We have very little understanding of science in the political arena. And again, that's a really big issue. And just to show you how easy it is to pull the wool over people's eyes, there was a study done by an American university asking people whether they were in favour or not of there being DNA in food. And they actually came up with a warning that they printed on food to tell people what DNA was. This is all entirely true. DNA is linked with disease. You can pass it on. And most Americans are saying, you're right, we should ban DNA in food. It sounds very, very dangerous. And you actually had a majority of people agreeing that this labelling should appear on all foodstuffs. Of course, virtually everything you eat has DNA in it, and it's a normal part of producing food and consuming food. I'll talk about GM crops in a minute, but it just shows the level of difficulty you have and how easy it is to generate conspiracy theories around the kinds of foods that we eat. The problem we've had as science communicators and science journalists is what we call the deficit problem. We used to think that the big problem in science communication was that people just didn't know about the science, and if we told them about the science, everything would be fine. So all we have to do is get out there and tell them the science that we're doing. And then they will be supportive, and we will have lots of support for science and lots of people interested in science. Does it work? Of course it doesn't. People don't operate in that way. That's not the way people understand science. The deficit model, this idea that we have the knowledge and we're going to share it with you and you will understand that knowledge, no matter how engaging you make it. I'm not going to give him up, but there is always a barrier there. And that's a barrier that you really have to struggle to overcome. So the deficit model doesn't work. We talk a lot more these days about engagement and interaction with people in terms of having conversations around science, but we have learnt over the years that the deficit model does not work. We cannot just tell people about science and expect them to understand it. So here are some examples as to how that's been working. Vaccine hesitancy. Now, I've been reading in the news that since Trump got elected, he is threatening to put a vaccine skeptic in charge of the US Health Department. When I was a kid, everybody got vaccinated, and my parents could remember when people used to get polio, when people used to get tuberculosis, and there was masses of support for mass inoculation. We've forgotten that. We've forgotten just how common these diseases were, and now we're seeing a huge rise in what we call vaccine hesitancy. A lot of that dates back to a paper in 1998, which was Andrew Wakefield's paper on the link between the MMR vaccine, that's mumps, measles and rubella, was linked in his paper to autism. Now that study was completely debunked, but not until it went all over the media, and it got a lot of traction. A lot of newspapers covered it. Link between autism and MMR. This was on the back of a number of medical scandals that had come out in the 1960s, the most famous one being thalidomide. This was a drug that was given to pregnant women to deal with morning sickness. It created massive deformities in babies. But it was a long time before people actually recognised that and had to deal with it. But the fact that this was promoted by big pharmaceutical companies was generally building this air of scepticism around medicine. And the problem with things now is that once you've got a scepticism out there, it gets amplified and amplified and amplified. So famously, Joe Rogan, who's also backed Trump, is a vaccine skeptic. You're seeing more and more people coming out. This was obviously a big problem on the back of COVID, whether vaccines worked or not. Now it is quite common for people to refuse vaccines for their kids. And we're starting to see some of the diseases we thought we would never see again in the UK coming back. So you're seeing measles coming back, you're seeing measles coming back in the US. So it's becoming a massive, massive problem. And it's because our perceptions of risk are very low now. Again, people can't remember these diseases. And the conspiracy theories just grow and grow. So here is one example where we are putting out lots and lots of information, get your kids vaccinated. People are just resisting it. So again, it's the problem of this deficit. Another example is the area in which I work, which is GM crops. Now, GM crops, genetically modified crops, are where you're taking the gene from one crop and putting it into another to give you a beneficial trait of some kind. So we work on a crop called camelina, which is an oil seed. We have put into that oil seed a gene from an alga in the sea, which produces a fatty acid called omega-3. If you've been in Holland and Barrett, you'll see those jars of omega-3 tablets you can take. So it's very good for your health. But most of the omega-3 that we use comes from marine fish. And we know that the fishing in the oceans is unsustainable. So we need to find ways of producing omega-3 oils in a less destructive manner. The way of doing that is through a GM crop. We're actually taking that gene, putting it in an oilseed plant. We can now we can grow omega-3 oils on land. We can't grow it in the UK because we're not allowed to. So we're growing it in the States, we're growing it in Chile. But why is there such a resistance to these crops, which could potentially be enormously beneficial, cut down use of pesticides, cut down use of fertilizers? Why are we seeing such a problem? I think part of the reason in the story of GM crops was the way that it initially came into the world, which was with companies like Monsanto engineering crops in ways that were beneficial to farmers, but also in ways which would boost their bottom line and make them money. So the classic example was a crop they created called a Roundup Ready wheat. What that means is there's a weed killer called Roundup, essentially it's glyphosate. They managed to engineer a crop that was resistant to that weed killer. That meant you could plant that crop, spray the weed killer onto your field, and you would get rid of the weeds and the crop would continue to grow. So actually it worked very, very well, but people took a look at that and said, well, that's just all about selling more Roundup. So again, the conspiracy theory starts to grow. The companies, the corporations are up to something here. This happened at exactly the same time as something called mad cow disease, which you may have heard of. So this was a disease of cows, bovine spongiform encephalitis it was called. And cows basically started staggering around and acting in strange ways. And we believe that cows actually got that disease from eating essentially the minced up remains of other animals. So it's quite common to, for instance, if you had carcasses of sheep, to mince those up and put them into cattle feed. So cows got infected. We were told by the government that there was no way that this, it's actually a prion, it's not a virus, this small infective thing could move from cattle into people. We were told that was just impossible. Turns out that was wrong. There started to be a handful of cases appearing of people actually developing conditions linked to the BSE in cattle. Absolute crisis for the farming industry. British exports of beef almost ground to a complete halt. But again, trust in the food system was being undermined. And people started hearing about things that other companies were doing, such as the Terminator gene. Now, this was a gene that was bred into GM crops, which meant that they couldn't reproduce. So you couldn't collect the seed from the crop and plant it next year, because the gene would stop it growing again. Now, the reason that was done, it was actually something that the companies were trying to do. The reason it was done was because people were quite concerned when GM crops first appeared, and they didn't know whether there might be an environmental problem from a crop. So companies were asked to include a Terminator gene so that the crop couldn't spread, just in case something went wrong. It was never deployed, but again, it seemed to feed this narrative that something was afoot here, something was carrying on that we weren't too sure about. And we had a lot of popular culture that was anti-GM. So this was a TV series called Fields of Gold in the early 2000s. That's a very, very young Benedict Cumberbatch on the right there. This was a mainstream BBC One primetime TV series about a leak from a GM lab that goes crazily wrong. Terrible things happen. There's a massive cover-up. It just fed into the narrative that something was deeply, deeply wrong with the GM system. There were some attempts to try and put this right. The government has tried to have lots of public debates around GM, but even now we still see a lot of pushback. So this is an area in which we're doing a lot of science. We think it can bring in a lot of sustainable traits into crops, but we still have massive pushback from activists who are anti-GM. We see them targeting us on a regular basis on social media. They are actively targeting politicians to try and roll back legislation that would enable us to develop some of these crops further. We're not asking for a regulatory-free world. We're asking for scientific-based proportional regulation. But the pushback is there. You will see on products in this country, this product is GMO-free. Very common. It may be not. We've got a lot of GMOs in our system already. Virtually all our animal feed is from GM-derived soy. Many things that we consider to be GM-free are not. And we still have people who are feeding this narrative that anything to do with GM is wrong. A celebrity tends to have a lot more resonance with the public than any scientist does. So, despite the fact that GM has proven very, very useful, I've put up two examples there. So GM rennet. Rennet is what you use to make cheese. Any cheese that claims to be vegetarian is a GM product, because that rennet comes from a GM-altered bacterium which creates the rennet, which is a good thing, because in the past we used to get rennet from calves' stomachs. So we ate a lot of veal. Calves were basically caged up. It was quite a cruel practice, and we used the stomachs of those calves to extract the rennet, because calves have that in their stomachs to essentially process the milk they get from their mothers. So all cheese was made with this substance derived from calves' stomachs. So much, much better to be able to create that from GM. But again, it is a GM product and it is in the food system. And virtually all the insulin that we use now for diabetes is produced through GM. Again, it's a huge benefit in terms of animal cruelty, animal welfare rather, in that in the past you needed large numbers of animals to create the insulin that you needed for all the diabetics. Now we can do that with a bacterium because we've engineered it that way. So again, GM, fine for insulin, okay for rennet, but people are very, very concerned about having it in their food. Just another example as to how it can go terribly, terribly wrong. Climate change. Trump, as we know, is a climate skeptic. Kemi Badenoch calls herself a net zero skeptic. Again, huge amounts of pushback, gathering momentum now as we move into the next climate COP in Baku, which I think started earlier this week. A lot of people are trying to ignore or push aside climate change. The scientific evidence and the scientific consensus is overwhelming. There's very, very few scientists who would question it. They may say, we're not quite sure what is going to happen. And I think that's fair to say. We don't know if certain ocean currents are going to switch off. We don't know whether we might reach a tipping point with sea ice in Antarctica or in the Arctic. But we're pretty sure something's going to happen because we can see many of those changes already. But the problem with communicating climate change as a science is, again, it's not an easy story to tell. We can't just point the finger at a certain group of people and say, this is where the problem is, we're all responsible for it. It works over a very long time scale. Basically, throughout my career, there have been stories about climate change. From when I started doing media with the BBC, quite a few decades ago now, we were doing stories about climate change. We're still doing them. And there are still people saying, well, we're really not sure if this is happening. And we've had incidents like ClimateGate, which have just added to that uncertainty. Now, ClimateGate was basically just before the UN meeting that's on climate change in Baku now. There was a meeting in Copenhagen in 2009. Just before that meeting, a whole series of emails were leaked from UEA, which has a very, very active climate research department. Those emails showed normal science in action. They showed how people manage data, manipulate data, yes, but the way that scientists do manipulate data, so you often ignore outliers, for instance, thinking, well, there must be something wrong in the way that we're measuring things. But the way those emails were presented when they were leaked was that the scientists were up to something, that they were manipulating the data, that they were doing things they shouldn't have been doing in order to make the case for climate change. When that hit the news about a month before the climate change conference, it basically blew any deal out of the water. And from then on, it's been a huge struggle to try and get back on track. We've had the Paris Agreement in 2016, which has made things a little bit better. But ClimateGate was a massive, massive blow to climate science. And it just showed how difficult it is to try and communicate how scientists work. Scientists embrace uncertainty. They embrace a little bit of fuzziness around the edges. They have to, because very little science is exact. You have to be a skeptic to be a scientist. So ClimateGate was just showing that the way that they used their data to try and get a result that was meaningful. But the way it was presented made it look like they were up to no good, and perhaps just fudging the results so that they could get something that they agreed with. And there are many, of course, well-funded vested interests who are pushing conspiracy theories about climate change. There's quite a lot of evidence now that some companies in the energy sector have been involved in this kind of climate denialism, but I think we are moving on from that, but that is still out there. And of course, on a personal level, most of us don't want to change our lifestyles, which makes it even more difficult to get a message across. So you're saying to people, the climate's going to change, you're going to have to stop eating meat, you're going to have to stop eating dairy, you're going to have to get rid of your car. People say, I don't want to hear it. I don't want to hear it. I'm going to carry on as normal, it's fine. So again, when science challenges your worldview, it can be enormously difficult to communicate that science, and makes it very, very challenging again for scientists to try and get their message across. And ultimately it comes down to a lot of people saying, well, there's nothing I can do, so I won't bother. And very, very difficult for scientists to deal with that, and the conspiracy theories again are abounding. I do believe there's more we can do about climate change communication. It is an area that we work on, working in the food and farming area, because it's a massive part. About a third of our global emissions are probably coming from the food and farming sector. So that's everything from changing land use, i.e. clearing forests, draining wetlands, to the way we produce food, the way we move food around the planet. All of these things are creating a big, big chunk of carbon emissions. As part of what we're trying to do with our research is to try and deal with that. We do think there's more that can be done. We have seen some amazing stuff in terms of the way that environmental destruction has been portrayed, particularly on television. That Blue Planet scene of, I think it was an albatross, sicking up bits of plastic for its young to feed on, was hugely, hugely influential in challenging online debates about environmental destruction. We need a lot more positive action. We need to show a lot more certainty. And again, this is actually quite a problem for scientists when they're trying to communicate. So our GM scientists, one of our leading professors told me that when he is asked now by a journalist, are GM crops 100% safe, he says yes. Now, he knows that is not true. But if he says, well, immediately they're going to say, leading scientist casts doubt on efficacy of GM crops. So it's not something scientists want to do or ever feel comfortable doing, but he feels this is the only way he can try and get his message across that what he's working on is beneficial. So an enormously difficult moral decision on his part. Should he tell a half-truth to get the point over, or should he tell the actual truth, which is that within the bounds of probability it's a yes. So very, very difficult for some scientists, but we somehow need to get better at communicating certainty. Science communication, well, I've mentioned that the deficit model doesn't work. What normally is lacking from scientific communication, one of the things that we're very, very bad at, is talking about people and talking about what motivates them. We often think that the science speaks for itself. I'm afraid it doesn't. We have to know more about you as a scientist. We have to know more about the kind of person that you are and why you're doing that work. It's vitally important with science communication. So we often talk to scientists, we do little video clips with them about why they're doing it. Because it's very easy in the abstract to say, well, we think all GM crops are bad. But if you're talking to one of our scientists who's really, really trying to do some positive change, create something that will have a massive environmental benefit, maybe that will change your view. So we want to connect with people. We need to put people back into science communication. I think there's been a tendency to take scientists out. I think that's very characteristic of the way scientists work. They're very self-effacing people, of course, normally, but also they want the work to speak for itself. They want the data to speak for itself. And I'm afraid we just can't do that. We need to put people back into the picture. So, the news. I used to work in the news. Scientists are often extremely frustrated about the way that science is covered in the news. But part of the reason for that is that the news editor sitting in the newsroom probably is not someone with a science background. And they're looking for things that are going to resonate with their audience. And these things, what we call the news values, are the things that drive what goes into the news every day. So when the team is sitting down, we're looking for stories that come into that. Very often, science does not. And that's why it is extraordinarily difficult to get science into mainstream news bulletins. Again, we have to work very, very hard about how we pitch stories to the media. We take those basic news values and we try and craft our press releases in ways that we think will resonate. And again, it's about human interest, it's about people. Often there might be a personal story about the scientists themselves or how they came to that point in time, what motivated their career, why they took the decisions that they took. And again, that can help us market stories. But we have to work within the way that news works. And unfortunately, news is driven by this agenda. It's quite well recognised. This is what journalists are taught when they're doing journalism courses, and we need to be aware of it. A lot of science journalists are now getting so frustrated with the way that science is reported that they're moving to a different way of reporting science, and it's called the truth sandwich. And the reason it's got that is because if Joe Rogan stands up and says, oh, all vaccines are rubbish, they don't work, and in fact they can be poisonous and deadly to you. If you report that on the news, Joe Rogan says vaccines don't work, that's what everyone's going to remember. So now we have to take an approach which never existed when I started in journalism, but is being recommended by a lot of science journalists, called the truth sandwich, which is that vaccines work. We have seen rates of disease fall to near zero since the 1960s. Today, Joe Rogan said they didn't work. But as far as we can tell from all the data that we have, vaccines work well. So essentially, if someone puts a bit of disinformation out, you sandwich it with the truth. It actually goes against the grain for me, having started in journalism, because we never used to work like this, but more and more science journalists are finding this is the only way that they can stop the disinformation from spreading. There is so much out there now that you need to have the truth sandwich, tell them the truth, tell them someone says they're a bit sceptical, tell them the truth again. So again, using quite clever techniques, maybe even cynical techniques, to try and get our message across is vitally important. And finding ways that resonate. We have seen that when we've used this approach, we actually, audience research from, particularly with PBS in the US, has shown that it's much, much better at trying to dampen down conspiracy theories and anti-science messaging. So it's well worth looking at. So just a bit about what we do at Rothamsted. we use all the main forms of media, public engagement, like Mark was talking about, a lot of face-to-face work, doing events, doing music festivals even. We work with the mainstream media. We do recognise they're still very, very important. Obviously, we run a website. Obviously, we put news stories on our website. The difference between me posting a story on our website and it going, say, on the Guardian website is about a factor of 100. So I will get, 2,000 or 3,000 views if I put it on our website. That will magnify up hugely if I get it on The Guardian and then be shared throughout the world. So very important that we still work with the media, even though we're hearing more and more talk about the mainstream media losing its importance. It still is a source of credible information for most people, and we still need to take it very, very seriously. And of course, we use a lot more social media. That is becoming increasingly important for content, but again, you've got the big, big problem of all the false information that's being put out on social media. And how do you know what is true when you're browsing through your social media feeds? How can you tell what's a reputable source or what is just misinformation? We know that social media is extremely important. You were talking about using Twitter. X, as we now have to call it, and Blue Sky, which a lot of our scientists are now much keener on. The evidence that using social media in a science career works is very, very compelling. We do know that it works very, very well in terms of if you're a young scientist, getting your work out there on social media builds communities, builds collaborators, great way of getting your messaging out, but there can be a cost. So, colleagues of mine at an institute called the Pirbright Institute, which is in Surrey, they basically work on animal diseases. And during the pandemic, when the pandemic first started, somebody spotted that the Pirbright had taken out a patent on a coronavirus. Now, actually, it wasn't the coronavirus. Coronaviruses are a family of viruses. It was one coronavirus they're looking at in chickens. And they had been working with that. That project had some funding from the Bill and Melinda Gates Foundation. Bill Gates, coronavirus, research institute, an almighty storm basically blew up around that. Bill Gates was trying to infect us all with coronavirus for some evil plot to get us all to use Microsoft more, and this just went stratospheric. They were inundated with trolling emails for weeks. Now, the comms team, who I work with a bit, could deal with it, but staff were opening up their emails every morning and opening up their social media feeds, more importantly, and getting a torrent of abuse. And you can imagine how damaging that is to you if you're a practicing scientist. So, social media comes with a cost. There's some very, very active click warriors out there, and whilst it's really important to get your science out there, sometimes it can go wrong. So, I've talked to you about all the things that can go wrong in science communication, but actually lots is going right. We have never had such an incredible amount of really high quality information coming out of science and science media. And it's wonderful to see. It was almost impossible outside of a few very niche programs when I was a kid to get science on mainstream media. Now it's everywhere. We've got lots and lots of channels dedicated to science. You've got lots more choice. But what could you do? What could you, if you're interested in science and interested in talking about science, what could you do? So you could do what I did, which was science journalism. There are plenty of ways of getting into that. I think it's really important that you do some science first. Most people who go into science journalism, like me, did a science degree first, and then decided to move into talking about science rather than actually doing it. That can be really, really valuable. There's lots of schemes available if you're a practicing scientist to get more information about the media. For instance, the Royal Society Fellowships, if you're a practicing scientist, they will pay for you to go and spend 6 months, say, working on a newspaper or working in a newsroom to try and learn about how they do science, to see how the other side works, as it were. And there are opportunities, but this is in decline. So there are fewer and fewer specialist science correspondent jobs available. Almost all traditional media sources now are challenged for funding. Obviously from the newspapers, they've lost most of their advertising. For things like the BBC and commercial television, again, budgets are very, very small. So there are less and less jobs. And that's a bit of a problem, and there are less and less science specialist posts. Now, there was a time when you would have a science journalist in almost every newspaper. People like the Daily Mail would have a science correspondent. Now, most people are probably just processing press releases, what we call journalism. They get a pile of press releases in the morning, and they write them into stories, and they put them on the website. And so there's very little quality control, very little trying to sort out what science stories are interesting, what they can actually promote, and it can be very, very challenging now to find dedicated science roles in a lot of mainstream media. They're still there, and there are still a lot of science journalists, but it is in decline. I think those opportunities will still exist by the time you finish your university degrees, and it's certainly worth considering. There are other things you can do working in science engagement. There are many science centres, there are travelling science shows. They almost always involve large amounts of explosions, but they're extremely popular. I know a number of people who've gone down that route and got very much essentially into a stage show for science. And again, well worth considering if you're going down the science road and you really want to promote what you do. And there's the kind of work that I do, which is working in communications or policy within science. Now, I do it because I'm really interested in science and I want to get the science out there. I want it to make a difference. But there are all kinds of things you can be doing in the world of a communications office. So the British Antarctic Survey have started producing these wonderful TikToks done in the style of Wes Anderson. I don't know if you know his films, but they're beautifully, go take a look on TikTok and find them. They're absolutely beautiful and very, very cleverly made. I put up there the Large Hadron Collider rap. That's pretty old now, but basically the young communications team decided to do a rap song around the Large Hadron Collider. It's pretty awful. I'm no expert on rap, but it was pretty bad, but 2 million hits. And when you consider that most science communication, you're talking in terms of the thousands, it was a great way to try and showcase what was going on at the Large Hadron Collider. This is a big facility in Switzerland. And then things like the Blue Dot Festival, which is at Jodrell Bank, which is basically creating a huge science experience around a major piece of science infrastructure. Whoops. So lots of things you can do, working in policy, working in communications, ways of taking science and making it exciting and interesting. Or you could do it yourself. You don't have to rely on mainstream media, you don't have to rely on a press office. You can set yourselves up as science communicators. So a good example is IFL Science. I cannot tell you what IFL stands for. You'll have to go and look it up. But it's not something I can say in public. That was set up by University of Sheffield science undergraduate. She felt science communication wasn't going anywhere. She set up this website with interesting science facts on. It's now basically a global brand. The Naked Scientists is very, very similar. Scientists at Cambridge wanted to start their own podcasts. Now, again, a massive brand. And then people like Greg Foote, who's a science presenter, set up his own channel on YouTube and uses that. Now, I guess you'd probably be talking more TikTok, but you can just do it yourself. You don't have to rely on the mainstream media. It's one of the beauties of social media is we now have access to production ourselves and we can put our own messaging out there. Lots of jobs in terms of science, TV and podcasting. It's all over the place at the moment. I've done quite a bit of this work myself. The research jobs are great. You get to find out about all different kinds of science. We need more and more people with science backgrounds to get into these roles. Endlessly, when you're trying to pitch ideas within mainstream media and you're a scientist, people are just going, yeah, that's not interesting. And really, really have to work hard to get people to accept it. I think that's changing. I think the technology we have now in terms of the CGI that can create, you look at Brian Cox's series on the universe and on the solar system, the CGI creating those new worlds is unbelievable compared to what it was a few years ago. And the camera work and things like Blue Planet, amazing. So we have got the technological revolution to enable us to tell stories. We just need more scientists taking the reins and getting out there and getting involved. And there are courses you can do as well in terms of science communication. Once you've done a science degree, it's well worth exploring the various institutions that do different kinds of communicating around science. You can also do, as some people here have done, incorporate science communication into your career. and there are quite a lot of quite high-profile scientists who have done that. They work as university professors, they still do research, but they do science communication as well. That's Ben Garrett at UEA at the top there. That's Helen Chursky, Jim Al-Khalili, who's a professor of physics. You can combine science communication with a career, but I still hear reports that some professionals find that colleagues in their departments push back on this, and that it's not taken seriously. So I like to think that age is behind us. I think it was quite common when I was younger, but I still hear occasionally people saying, well, it's just flim-flam, it doesn't matter. I think it really does matter, and I think it's really, really important for all scientists, particularly public scientists, people in publicly funded institutions, to get out there and try and communicate their science. Maybe you don't want to stand on a stage, maybe you don't want to do a podcast, maybe you want to write a blog or create a video game, whatever it is. There's plenty of opportunities. So just to finally say, there's a huge, huge range of things you can do to get involved in science communication. We need more talented scientists getting involved in communicating the science. I think it's central to what a scientist should be, is to get out there and talk about what you do. It has never been more important in an age of so much disinformation, conspiracy theory and anti-science for people who love and know about science to get out there and to make the case for what just can be a hugely fulfilling and life-changing experience as a science career. And that's really my final point, which is whatever you do with jobs, if you're working in science, try and find a way to communicate what you're about and communicate what you're passionate about. There's always a way you can do it, even if you don't want to stand up on stage. So think about how that might relate to what you're doing, and try and make a difference in trying to make sure that we get the message about how positive and wonderful and life-enhancing science can be. Thank you.
Has a professional scientist that worked with you ever leaked information? So the ClimateGate in 2009.
How did ClimateGate happen?
So no, has any of your colleagues leaked information from? Have
we been the victim of that kind of trolling? Well, the main example I told you about, which was the coronavirus thing, would have been the main thing I've been involved with, where an online campaign just builds, and as a communication professional you can deal with it, but for working scientists, having that abuse heaped on you is really, really damaging. ClimateGate, there was a terrific, I think it was BBC, a terrific drama series about it, which may still be on iPlayer, I don't know. But the scientist who was the head of that particular department had nearly destroyed his life. And he nearly committed suicide because of the amount of pressure that was put on him and the accusations of scientific malpractice, simply for doing what he normally did as a scientist. And he's quite a mild-mannered, self-effacing man. If you want to learn more about this and the risks and the ways to go about it, there's a terrific book called The Geek Manifesto by a guy called Mark Henderson. It's a very easy read. He's a non-scientist who was asked to do the science correspondence job at the Times, which just shows you where we are. But he really bought into it and wrote an incredibly good book about why science communication matters and how it can go wrong and why you need to get out there as a scientist. So I'd certainly recommend that as a way of doing it. But yes, a lot of scientists in very public roles are feeling increasingly threatened by the online mob now. And it is quite challenging, and we, as communicators, we try and deal with it. But when people are personally targeted, I think that's very, very damaging. We have strategies to deal with it, but if you're working in a very controversial area, you can expect it now.
Okay, thank you.
Hi, so for the field communication part, I have a problem about, I have a question about the self-efficiency, so I didn't really understand what that meant.
Sorry, this microphone.
Self-efficacy. self-efficiency.
Self-efficacy.
Efficacy, yeah, I didn't understand what that meant.
Oh, sorry, yeah, sorry, didn't explain that. Yeah, self-efficacy is a belief that you can do something about a problem, that you can change, that you can do. We use it a lot. I work quite a bit in public health, where messaging is very, very difficult. But self-efficacy is a belief that you can actually make a difference in your own life. So I worked basically in the early days of HIV in Africa. I worked on public health campaigning out there, and a lot of it was people saying, "What can you do?" You know, it's in the hands of God. Self-efficacy is say, I can do something. I can change my behavior. I can make a difference. So that's what that's about. Sorry, I should have explained it.
How do you go about communicating statistics to the public? Because statistics is quite a difficult thing to be able to communicate.
Yeah, well, yes. I mean, you know, I did put up that thing about risk and statistics and probability. These are the most difficult things to communicate. We have, there's a mismatch between the way we've evolved and assess risk and what statistics tell us. And that is very, very difficult to overcome. So, you know, so it takes something like nuclear power. Very, very unlikely that it's going to affect you, but it only takes one Chernobyl for everyone to go, my God, this stuff is dangerous, or Fukushima would be the other example. So I don't have an answer for you, but I totally understand the problem that statistics are a massive issue. So that what I told you about, what I mentioned earlier about one of our senior scientists trying to be yes, no binary when he's asked questions about his research, he hates doing that, but it gets misinterpreted if he just says anything else. He has found a way of saying it, which is on the balance of probability, I can tell you it's 100% safe. But statistics and the nuances of statistics are enormously difficult. If you've got an answer, I'd love to hear it, because we struggle with it. And we're often, when we're putting out a press release, we're looking for the number. You know, a third of all agriculture could be changed to not use pesticides, say, a sort of thing we'd work on. But actually the statistics will be, you know, there'll be confidence levels and there'll be, you know, a massive range of figures. We pick the number that we think is going to work. It's not ideal, but yes, you're quite right. It's a very, very difficult thing to communicate.
Sorry, not a question, but a plug for another forum for communication. But people I worked with a while back at Imperial College, junior researchers, postdocs and PhD students started up a group called Pint of Science. The idea that junior people can get involved and communicate science to their local communities in a pub initially, but they've moved online. They've got lots of forums in London and also in Sheffield, so it's worth looking up. You can find them as a presence online. Thank you.
Oh, that's long. So do you know earlier you were saying about how they leaked the emails, and it was literally just about manipulating data. Obviously, if you do science A levels, you'll know what that means because you do it in class. So do you think how you're saying there's a miscommunication, would you, or your field of work, do you help maybe put that forward to younger, what am I trying to say? Younger, in education, younger years, so that could, that sort of data could actually be understood more?
I mean, it's very difficult because you have to, in science you do manipulate data. It's one of the things you do. Do we tell the younger scientists not to do it? Is that what you're saying? Or?
No, I'm saying, do you think, no, I'm agreeing that everyone should know what that means. But I'm saying, do you think it should be introduced in younger years of science, what that means? Because in GCSEs you don't necessarily learn about that, but in A-levels you do. So if we let's say read the email,
talk to your teachers. We have to talk to your teachers about whether you can manipulate your data or not. Is that maybe they'll, I think part of the problem is that word, manipulation.
I was just generally saying a general sense, not just the topic of manipulating data, just generally how science actually goes about stuff.
I think there's no way around the fact that when you get data it can be messy and you have to try and impose some sort of order on it in order to get meaning out of it. We're much better at it now with things like AI and machine learning are really transforming the way we interrogate data. and it is giving us patterns that we could never see before. There may be a way through with those kind of technologies in actually saying, well, this isn't a human thing, we've trained the machine to look at the data, and that's how it's coming out. The problem with the climate gate thing was the way the manipulation was taken out of context, and little bits of the emails were published. It was very, very cleverly and malevolently done. And there are lots of suspicions as to who might have been doing it. But that was I mean, people often say to you in communications, never write anything in an email you wouldn't want to see splashed over the cover of the Daily Mail, which I take to heart to a certain degree, but not about some of my colleagues. It's difficult. I don't have an answer for you, really. Working with data is difficult. You learn how to get better at it. I do think the word manipulation has some very unfortunate connotations, but it's the word we use.
Yeah, thanks for a really interesting talk. I mean, I agree with you at the outset about the inability of scientists in the past to communicate without, as you say, getting away from the probability aspect. But obviously you touched on the growth of social media and how much miscommunication goes through that channel. But obviously we've now got Trump about to come back in the White House, who's not the most scientifically literate person on the planet. He's also out to get journalists. How pessimistic are you about Trump dumping on scientists, basically, as we go into the next 4 years?
I think that's very, it was weird. There was I saw a tweet from Richard Dawkins this morning saying, he was worried about Trump's anti-science agenda, but it was fine because Elon Musk was going to be in the government as well, which I'm not sure I totally agree with. I think it's really difficult, isn't it? I mean, we don't know what he's going to do, frankly, but I think the whole message that science, the whole anti-science tone of what that administration is kind of promoting, I think it's worrying. I certainly know, looking at our feeds, that American scientists are increasingly worried about what this means. We will carry on, of course, doing what we do in this country, but I think, Americans are brilliant at science. They have more Nobel Prizes than almost any other country. They are some of the best scientific institutions in the world in the States. I think it's very worrying to have an administration that might not support that effort. So it's 4 years, we've got to hang on. But yeah, very worrying.
Sorry, I had a question kind of referring to what the young lady was saying there. So at the moment, science is going under all subjects, according to the government, are going under a curriculum review. And I was wondering, following on what the person said over there, are there certain words like manipulation, I don't know, data analysis, all of this, that you would like to have a key set of definitions or something like that, so teachers actually say this is what it actually means in science? I'm not sure I would go that far, but I would say I think it would be important to talk about science communication within, and this is an age of misinformation about science that we have never seen the like of before. I've never experienced anything like it, and I think it's really important that everybody, not just people studying science, gets more training about at school about trying to work out what is true and what is not, what scientific evidence means, and when something is backed by scientific evidence, what that means. And it doesn't mean something is a certainty. And communicating that, I think what's important is that everyone should learn that at school, because we've never seen the tidal wave of misinformation that we're getting now. And it is quite worrying. People just need those critical thinking skills. How you teach that, well, I'll leave that to teachers, but I think it's become something that's an absolute necessity now.
I'm afraid we've only got time for one more question before we have to move on. It's amazing that there are questions, and please do feel free to ask them a little bit later on.
I was just wondering, talking about science communication, we are trained as scientists, and in fact a lot of the posters at the back here, we're trained to communicate in a specific way, and that way is very obtuse to a normal person. Like, even for me as a scientist, reading some of these posters, they're outside my area and I'm confused. Do you think we should be spending more effort and that there should be more put onto career progression spent on trying to communicate with the layperson rather than just to other scientists?
I think that's quite difficult. I think it has to be a sort of coalition of the willing. Some scientists work in very specific, precise areas and probably don't need to communicate very much of what they do. Other scientists, I think it's very important that the public at least understands what the purpose of it is, even if they can't understand the data. Scientists, this thing they call the inverted pyramid, which is when you do a scientific research paper, you start off with the data and you work your way through and you end up with a conclusion. And the media flips that on its head. You have the big headline at the top and then you go down to all the detail at the bottom. So it's actually the inverse of the way that scientists normally communicate is what the media does. That's quite a challenging thing for people to get their heads around. But I think those scientists who are interested in communication, I would definitely like there to be more resources and more support for those people to get out there. But I totally accept it's not for everybody. Some people are going to be working on the minutiae of, you know, some obscure thing. Some things in science, frankly, are possibly uncommunicable because they're so difficult and technical. And I think that's okay, that only, you know, your direct peers know what that means. But someone somewhere needs to paint the big picture. And I think you're finding those spokespeople is going to be really, really important moving forward. People for whom there's credibility, so that when Joe Rogan stands up or Gwyneth Paltrow stands up and says things about vaccines, there's someone equally credible who can turn around and say, this is the evidence. Sorry, I've got to go.
Can we have a final round of applause, please, for James Clarke? Because you've all been sat down for so long, we've got a bit of a shift. So you've got about 5 minutes to stretch your legs, go to the loo, do whatever you need to do, and we'll be back here at 1:45, please. Hey everyone, can I please encourage you to find your seats for our final speaker, Vanessa Madu? Whenever you're ready. Ladies and gentlemen, please allow me to introduce Vanessa Madu to come and speak for the final time today. Thank you.
Okie dokie. Well, hello everyone. It is an absolute pleasure to be here speaking with you today. I am having a slight struggle with the clicker. Oh, can someone help? Oh. Never mind. So yeah, hello everyone. It's a pleasure to be here speaking to you all today. I kind of just want to get straight into it. You'll find out a bit more about me as we go. But the thing I can definitely say to start with is, hi, I'm Vanessa. I am a mathematician, I am a statistician, and I am an ocean scientist. But above all of that, I am a strong advocate for the idea that science lectures should be interesting, they should be curious, they should be whimsical, rabbit hole inducing, insightful, and above all, fun. And so I'm going to try and use my time speaking to you all here today to do that. And the way I'm going to do that is at some point in this talk I'm going to spontaneously burst into song. I'm going to show you my rubber duck collection because I have a quite extensive one. I am going to geek out about my research to you because you're a captive audience and that means I can. And I'm going to spend the rest of the time discussing some ridiculous hypotheticals. But first, the story. I come bearing a tale of whimsy and adventure, and these are the stories I love to tell. Today's story starts in the heart of the sea, amidst strong winds and a large ocean swell. A ship in the Pacific signals distress, not for danger or peril, but because of a very large mess. And though I'm a scientist that is academic most of the time, my preferred means to share is through rhythm and rhyme, if you'll allow me. 29,000 ducks went swimming one day over the ocean far away. The shipping crew cried, "No, wait, come back." But none of the ducks came swimming on back. This accidental release of yellow floaty friends was the talk of the town in 1992. And while many chuckled at the novelty of it all, the oceanographers of the day, starved for data, saw an opportunity. The data is data, whether it comes from expensive ocean drifters or humble rubber ducks. And the journeys that these rubber ducks took helped us to understand the currents in the North Pacific and across the global ocean. And as a mathematical ocean scientist, this is pretty much exactly what I do, albeit with a slightly more sophisticated kit. The ocean is a powerful engine moving vast amounts of water across the globe. A rubber duck in the North Pacific could find its way to the shores of Galway, riding the ocean's superhighway. If you want to know where things like washed up or washed aboard rubber ducks or Lego, or more importantly, things like oil spills or marine plastic end up, then we're going to need data and a lot of it. The trouble is, the ocean is vast, it's massive, and it's opaque from the surface. We can't exactly take a telescope and have a look down there. So if we want to understand things about the ocean, then we have to be a bit more creative. But we do have a few options. We could pull a 1992 oceanographer style maneuver and use things that have been washed overboard, though that is suboptimal. We could use satellite data. Then there are drifters, which float, and floats, which sink. And then we've got gliders and more buoys, or buoys as the Americans say, which I quite like, and lots of other options. But I look at the surface. And so when we're looking at the ocean surface, there are a couple of specific types of data that are particularly useful. One being processed satellite data. So satellites move over the Earth. Some of you will have encountered this if you're taking physics. And they have these orbits that are polar, and they collect data. And then in order to make it a sort of nice, clean, processed product like that, we need to do some processing, and it takes some time, but gives us a big picture view of what the ocean is doing. And then there's drifter data. So drifters are funny pieces of kit because in order to capture data from them, oceanographers, seagoing oceanographers, I'm not a seagoing oceanographer, I don't like boats, but those who do will go out with one of these buoys, like up there, and throw them out in the ocean. And then they'll go on their merry way, being moved around by the currents, and then eventually, hopefully, we get to have them back. But in the meantime, it's transmitting information about what, it's finding where it is on the Earth, the salinity, the temperature, its velocity, and that kind of thing. And so we get these massive, massive data sets that kind of look all squiggly and a bit like spaghetti. And so those are the two types of data that I specifically work with. Then it comes to modeling it. So, oops. Okay, so when it comes to modeling the ocean, I look at a few different specific types of information that I'm particularly interested. There's this thing called geostrophic velocities, which are these large-scale ocean currents that happen because the Earth is rotating. If you think about the most pressing weather phenomena that happen on the Earth, they're things that spin, so cyclones, hurricanes, tornadoes, and things like that. Then also, if you've ever been to the beach, and on a windy day in particular, you'll know that the wind has something to do with ocean surface currents. They can really whip up a storm. And so I've also decided in my model I want to include information about the wind, so stress and speed. And then we've got position. So the ocean near the equator is going to be very, very different to the ocean in the North Sea. And then time of year, hurricane season versus not hurricane season, for example, tells us that the ocean has these really distinct cycles where the behavior is different depending on the season. So we chuck that into a model, and then out of it we get a drifter velocity prediction, and that drifter velocity prediction is as a probability distribution. This is kind of important, and I liked this more than I thought I would when I started my PhD, because it allows us to capture this idea of uncertainty. So cool, we make an estimate or a prediction about what a drifter is going to do, but how sure are you? And when it comes to presenting climate-related statistics or ocean-related statistics, that's really important. And so that's why we're opting with something like that. But the very observant of you might be sitting there thinking, Vanessa, what's in the box? And that is a good question. You shouldn't let me get away with that. In fact, you should ask other people that. When they tell you about a model, ask them what's in the box. So I will tell you what's in the box. But, and I promise I will actually tell you, I do need to take a quick aside first. I'd like to tell you another story. This will be a theme. So, some of you might have heard this before, but for the benefit of those of you who haven't, this is the story of the blind people and the elephant. And so we got 3 people, and they have never seen an elephant before. And they are blindfolded, so they can't see the elephant that is there in front of them. And so by touching the elephant, they have to try and figure out what an elephant is. And so one of them touches the elephant's trunk, and they say, oh, it's I know what an elephant is. An elephant is like a snake. And another touches the elephant on its flank and says, okay, an elephant is like a wall. And the third touches the elephant and touches the tail and says, okay, no, an elephant is like a rope. And this is a very well-known story, and I'm going to give you my rendition of this story. Instead, we're going to look at the decision trees and the ocean surface current, although now that I say it out loud, it doesn't quite have the same ring to it. So, same concept, where the decision trees, I ought to start with what those are. So they are a way where we can make predictions using data, so they're a form of model, and so you train them on data. So a common type is a classification tree. So if you're trying to teach a machine to recognize cats, then you might go along with saying, okay, if this image doesn't have ears, then it's probably not a cat. If it does, then you go to a new thing, a bit like a flowchart. And you can do this with numbers as well, so that you can predict velocities. And so each of these trees sees a tiny bit of the data, and then it recognizes whatever it thinks is useful, and then it makes its guess. But as you can see, these models still disagree with each other, and this is based on the parts of the data that they've seen. In the same way that the blindfolded people only have access to the specific data they have, i.e. the part of the elephant they're touching, the trees only receive a small amount of information as well. So, in terms of how it actually works, we start with the probability distribution. This is a very well-known bell curve. Most of you are going to have seen this before. This is a normal distribution. And so we start with one of these. Now, one of my trees comes along and it has a look, and we give it a small amount of the data, and it has a think, and then it decides based on that data that it's seen, I'm going to make this change to the model. But there's an important thing here about whether or not everything should be weighted equally. Because if we go back to my elephant analogy, if I've got a person that comes along and touches the tusk of an elephant, they might say, oh, I know what an elephant is. It's like a vase because it's smooth. But everybody else who's touching the elephant is going to turn around and say, absolutely not, this thing is wrinkly. And they're all right, but we'll need to weight the person who says that the elephant is smooth perhaps a little bit less. Not because what they said isn't true, but because it doesn't accurately describe the rest of the entity. And so, depending on how well this particular tree's contribution helps, we will weight it accordingly. And then we repeat again and again and again and again for a long time. Each tree giving us a little bit of insight as to a particular part of the data, and then we sort of throw them all together and get a consensus. And they'll be weighted differently, as I've tried to illustrate here. And then we get a distribution out. And so this is the sort of everyone's best guess, it's the consensus guess. And this is the kind of model that I've been using, and it looks like that. And that's, yeah, the sort of mathematical language equivalent. But it does say the same thing. So if you got my description thus far, you get that. So you can be pretty proud of yourself. And that is called multivariate normal gradient, natural gradient boosting. And so that's the really techie part of the talk done. This research is very much ongoing. It's something I'm currently doing, so you get to know some cutting edge stuff, which is pretty exciting. And one thing I would like to say is I'm really happy to take questions about this at the end if you're sort of interested about any of the techie bits, but I might not know the answer to your question. And that, if you're wanting to be a scientist, is a really important feeling to get used to. First of all, knowing that you're not always going to know. Second, being able to admit it. So I will absolutely give my best guess, or my best, I do know some stuff, I will give my best answer. But yeah, that's something I wanted to make sure that I got to tell you, is to be comfy with not knowing, because part of what makes science so fun is the fact that we don't know, because it gives us things to find out. But I'd like to take of pause for a moment, take stock on where we are in the agenda. So I have burst out into song during this lecture so far, and I have also, that tick is not in the right place. Oh well. I have also geeked out about my research. Next thing I'd like to do is a quick reminder. Scientists are also people, and people have hobbies. And here are some of mine. I decided I'd like you to all get to know me a little bit. I have lots of things I'm interested in. I think I do some really, really hard stuff during the day, so I do enjoy a sort of creative outlet. I crochet, I collect leaves, I also take care of plants. You're going to see a theme along the bottom there, various collections of reasons I really like plants. And then I draw and I like animals, but the main reason that this is up here is I do a lot of just for fun maths. And whenever I tell people that I do just for fun maths, I genuinely get a horrified look from people. The main reason for this is because I do maths for my job, so why would I want to do maths even more? And my answer is because it's different. One of the things I do is attend recreational maths conferences. This picture was taken not even 2 weeks ago. That's my rubber duck collection, so I've now shown you that too. And so the thing that we find that we do at recreational maths conferences, and the reason I like them so much, is they introduce you to the concept of maths as a play object. And that is probably going to feel extremely foreign to most of you. You would know maths in the context of I learn this thing, I do this thing. You know, you might have opportunities to explore a bit, but by the way that school works, you will be, you have to pass exams. And so one thing that's made the difference for me is being able to take a mathematical concept and see how far we can run with it. Looking for edge cases, basically. How can we break this? And so we will be doing a little bit of that towards the end of this talk. But while we were talking about hobbies, I also really like Alice in Wonderland. You will be able to tell that from my dress. And so, in order to do the next bit of my talk, I would like to take you to the land of cards. All right. So, our story unfolds in the land of cards. The Queen of Hearts has accused her dairy clerk, the Three of Spades, of tactful sabotage, and has demanded that he be relieved of his head. However, this is all based on a simple misunderstanding of chance. So allow me to take you to the courtroom. The Queen of Hearts, she made some tarts one chilly winter's night. She took a bite, frowned, and said they didn't taste quite right. The Queen declared, since rancid butter always tastes so vile, it is the dairy clerk that must be brought to stand accused at trial. However, the court did not agree with the Queen upon her stance, as the tart's bad taste could have come from flour, eggs, or salt. Many things could have caused the Queen of Hearts her woes, and because of that, the dairy clerk was finally free to go. So, what's happened here? The Queen reflects on the court's decision, which stated that even though bad butter leads to bad tarts, the presence of bad tarts does not mean bad butter. The Queen has committed a very, very common fallacy. In diagram form, she has much too much confidence, and she also doesn't have any evidence for this causal relationship that she has proposed. So she said, I know that bad butter will make pretty much anything taste bad, therefore it must be the person who's in charge of my butter, the Three of Spades. You know, she has other people in her vicinity. She has the king, she has the knave, who has a history of tart-related trickery, and then one of the members of the nobility, the Eight of Clubs. Now, when you become aware of this kind of thing, you start to see people making sort of incorrect causal inference or getting causal relationships wrong everywhere. It happens everywhere. So one of the things I'd like to leave you with is to see where else you can spot this, where two things are related to each other, but the order of what caused what has been mixed up. Anyway, to get anywhere, we need more evidence, and luckily, some transpires. So the Knave of Hearts was noted to be skulking around the grounds on the morning of the incident, and he had an unusually large bag. So the Queen has updated her position, so she is now less sure that it was the Three of Spades. Her suspicion of the Knave of Hearts has increased because she knows that this is a pattern of his. And on the morning of the incident, the King was with her. So her suspicion of him has decreased. Right, next. The royal stationers have decided that we will be using recycled paper moving forward, which is entirely irrelevant, uncorrelated, and shouldn't make a difference, so the Queen's position doesn't change. Lastly, an unscheduled delivery of butter was brought by a person unknown to the guards that morning of the incident. And the Queen, she knows that penmanship anywhere. That's the Knave's handwriting. And so with that, her opinion changes again, but notice this time her confidence that it's the Knave is not quite as high as it being the Three of Spades. She has learned that she can be wrong, and so she tempers her enthusiasm. And with that, you are all ready to go off and proclaim that correlation does not imply causation, which is essentially what I've explained here through slightly whimsical means. The world needs it. Speaking of which, we've not seen a globe in a while, so here's one. And on here, we've got the prime meridian. So, I don't know, how audience participation are you feeling? Oh, okay, I've got a thumbs up. Excellent. Does anyone know why the prime meridian is where it is? Yes. Yeah, it's a result of, so I'll just repeat that back. It was the centre of the British Empire. So Britain had a significant hold on the world's oceans, and so when it came to deciding how the ocean would be navigated, but also how time would be told, it was decided that the centre would be over Greenwich, and so that's why we've got why the prime meridian or zero longitude goes through Greenwich, and the corresponding time zone, GMT, where it is. See, I was going to read my watch, except I've got a stopwatch on my watch, so I can't tell you what the time is. But in any case, I spend a lot of time thinking about time zones because they're very mathematical. And so, well, let's talk about them. Now, if you are wanting to move around the Earth, or a sphere in general, to do that, you need some apparatus. Before that, if I wanted to move around this room, I could describe how I moved around the room via some steps forward, some steps to the right, and however I get up, I don't know, movement up and down. X, Y, Z coordinates. Now, when you're moving on a sphere, things work slightly differently. Well, you can use X, Y, and Z coordinates, but this is inconvenient. So there are other ways to do it. And so the way that we tend to do it, we do it on Earth, you'll be familiar with this, is starting with longitude, so how far round the Earth are you going, and that's going in the sort of parallel to the equator. Then you've got latitude, so going up and down towards the poles. And then, for the sake of completeness, you've also got the distance from the centre of the Earth. And those three things together make spherical polar coordinates. So have you met sort of regular polar coordinates? Has anyone? Okay, some of you have met this. So this is an extension to a sphere. If you're interested, ask your teachers later. But there is a sort of an interesting thing that I want to just bring up, so I'm going to go back a bit. So if 0 is there, and that angle is going to decrease, so you're going to have a negative angle on the left, positive angle on the right. When you meet the back, so directly opposite that 0, what happens? Because on one side, you've got minus 180. On the other side, you've got plus 180. And you've got to jump. Well, that line is called the anti-meridian. And that's at 180 degrees east or west. Now, I tied longitude to time earlier. So we can do that. On one side, we've got GMT minus 12. On the other side, GMT plus 12. And then the line where that 24-hour jump happens is the international date line. Now, the international date line, if left to mathematicians, would look like that. It would be the 180th line of longitude, to be the 180th meridian. But it's not left to mathematicians. So it looks like that. And so there's some very, very interesting geopolitical history here about why we've got sort of that hook in the middle. That's where Kiribati is, so that they could continue trading with New Zealand. Then, of course, Russia wanted to have all of its territory in the same time zone without the 24-hour jump. So you've got that very clear split between the east of Russia and Alaska. But this is entirely arbitrary. Humans decided that this is how we were going to tell the time. So it doesn't need to stop there. We can take this further, and I'm going to suggest that we do. So, in terms of a mathematically valid international date line, all we need to do is to make sure that there is at some point a jump between GMT minus 12 and GMT plus 12, so that 24-hour jump has to happen somewhere. And it has to happen at every latitude. That is really far over, but every latitude phi. And then we have to know where it is. So, some mathematically valid options. This would work. It might solve a lot of arguments. This would also work, and I think it would cause a lot of arguments. This would work, and I think it looks like a present. But my favorite one is this one. So, the algorithm for this is you start at the North Pole, and for every latitude, you ask for a random number between minus 180 and plus 180. And so that's going to be your angle where, that's going to be the point where the international date line is. And so you start there, and you go down, 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 down. And so what happens if you do that is the international date line looks like this. And, you know, if the international date line looks like that, you know, you could just choose your own, because really, who am I to tell you when you can recognize the start of a new day? So, to wrap up, I have burst into song to explain the 1992 saga of rubber ducks in the North Pacific. I have told you about my research in statistical ocean science and the models that we use. I have shown you my rubber duck collection, and we've discussed some ridiculous hypotheticals where we've attempted to break math slightly. And all of that kind of explains, or it nicely puts together why I like math so much, because it's both a vehicle for really serious science, but it's also a vehicle for some really ridiculous silliness. It is both work and play for me. So with that, we're done.
Are there any questions for that? Yes, plenty.
Hi, I know it's a bit of a stupid question, but do all your ducks have names?
That's not a stupid question. That's like my favorite question I've ever been asked at a talk ever. Not all the ducks have names, but a lot of them do. I have a Dracula-themed duck, and his name is Count Quackula, so I thought you might appreciate that. And then also, I've got with me my mascot, Pip. Yeah.
Which oceanic regions are you putting your drifters into?
So I don't do any drifter placing myself because I don't like boats. I'm also not a physical oceanographer. But in terms of the regions of the ocean I study, I'm currently looking at the northwest Atlantic. And so that's the part of the Atlantic Ocean that is off the coast of the East US. So sort of Florida and that kind of that kind of way. I would be interested in looking a bit more at the Southern Ocean. So the Southern Ocean, or just the global South in general, was sort of criminally understudied, because if you think about the global powers that exist in the global North, there's a lot of incentive to study there, and so a lot of the data is biased up towards the North, and as well as the study. So I would find it interesting to have a look at what's happening, because there are some really interesting features in the global South, especially the Southern Ocean, because the Southern Ocean is the only stretch of water that is completely uninterrupted, as you sort of, there's no land stopping it as you go round, and so it can sort of whip up to some really, really fantastic speeds. You can get some really rough and interesting seas there. So yeah.
Can we please have a final round of applause for Vanessa? The final part of the day before we get to the award ceremonies for the best posters is a Q&A with some of the sort of senior members of the YSJ. So all of today has been made possible by their tireless hard work, and they've been working behind the scenes, chuckling away upstairs. And this is a perfect opportunity for you to ask questions about how you can get involved in the YSJ, how your school can get involved, how you can get your research submitted. So if you're looking at getting your poster submitted to be published, this is a perfect opportunity to do so. But can I please invite the YSJ members to please come up onto the stage? See you.
like, what was the Young Science Journal like 5 years ago? And what do you think it's going to be like in 5 years?
just before we finish up, thank you so much for your patience today. You've been absolutely incredible, and the quality of your written posters has been superb. We have come up with 3 winners, one for each of the major science categories. So if you are called out, please do come to the stage, and please receive a little bit of a gift from us along with a certificate for the day. So for the field of biology, from the Judd School, Maximus Burwood, for their poster, Genetic Inheritance of Preference for Ethanol and Sucrose Solutions with Equal Caloric Content in Drosophila melanogaster. Isabella Furness from Denston College, with their poster entitled The Effect of Graphite Powder on Stain Reduction from Eggshells, a Simulated Tooth Model. And then finally, in the field of physics, from Sevenoaks School, Gabriel Lamb, for his poster entitled Classification of Astronomical Spectra Using Neural Networks. Finally, just thank you so much for today. A little bit of housekeeping before we make our way. If you would like to keep your poster, please do take it on your way out. Otherwise, if you have any questions for any of the speakers that were around, any of the panel that are around, we'll be here for the next sort of half hour or so. So please